Good morning, everyone. Welcome to the second day of International Conference in Trends in Chemical Research. I'm sorry, the session is running slightly late. There were certain changes in the schedule. So accordingly, we had to start 15 minutes late. Uh, for the information of all who have joined late, I would like to inform that this conference is being organized under the aegis of Department of Higher Education, Government of Haryana, and Shahid Udham Singh Government College is organizing this conference through the Department of Chemistry. This is the day 2 of the conference. Very eminent speakers represented their research work in various invited talks and in oral and e-poster presentations yesterday. In today's technical session 3, we have three speakers with us, three invited speakers with us, Dr. Anis Abrol, Dr. Manmeet Rawat, Dr. Jayant Sindhu. Then in technical session 4, we will have with us Dr. Sushil Pandey and Dr. Badri to share their eminent research work. Starting the technical session 3, first of all, first speaker will be there has been some technical issue. Sorry, my link had gone. So carrying forward the technical session 3, I would like to inform you that there is slight change in the schedule. The first speaker on list is Dr. Jayant Sindhu today, and the session will be chaired by Dr. Praveen Jangra. So may I know whether Dr. Jayant Sindhu and Dr. Praveen Jangra have joined or not?
Good morning, ma'am.
Good morning, sir. Welcome to this conference, International Online Conference organized by our college. I welcome you on behalf of the entire organizing committee. I welcome all the respected faculty members from different parts of the country. I welcome all the eminent speakers, all the eminent chairpersons. This conference is being organized by the Chemistry Department of Shahid Udham Singh Government College. The entire organizing team, I would like to appreciate you are putting in very good efforts to make the program a success, and it's going in flying colors. Dr. Praveen Jangra, have you joined? If you have joined, kindly let me know. So far, Dr. Praveen Jangda joined. I would like to ask any of the participants to share the feedback with us. Your experience of the first day of the conference, if anyone would like to share, is heartily welcome. Anyone from the participants for the feedback, please? Along with the Meet session, Google Meet session, YouTube link is also shared. those who are not able to join the Meet link, they may follow us through the YouTube link also. Gulab sir, Sushil sir, may I know the status whether Praveen Janga sir is going to join or not?
Professor Deepa, he will be joining shortly to us.
Praveen Janga sir is going to join shortly. We'll wait for 2 minutes. Am I audible? गुलाब सर आप बताएंगे am I audible or not?
Yes, ma'am. You are clearly audible.
Okay, thank you. if there is any further delay, whether we may proceed or not, will also kindly tell. Should we proceed?
Professor Deepa, our session chair will join in only 2 minutes, as
Okay.
our telephonic conversation with him.
All right. All right. So I would request the participants to kindly bear with us. So far, the session chair joins us. Meanwhile, anyone from the participants, I would like to come forward and share the experience of the last day and give the feedback. Anyone from the participants, please? Sumit sir, would you like to share your experience of the last day?
Yes, ma'am. It's really a well organized conference in history of the Department of Chemistry of our college. And really all the speakers were presenting their content very clearly. That was so much deep. It will take new heights to the research.
Well, thank you, sir. Thank you so much. Anyone else from the participants? You may come forward to share your feedback. Any suggestions from you for further improvement will be highly appreciated. Anyone?
Hello, good morning, Dr. Deepa. I am from Government College, Panchkula.
Hello, ma'am.
I
Welcome,
must
ma'am.
congratulate you for organizing such a good event. It was very
Thank you, ma'am.
surprising for me. I wasn't expecting this level of organization. I must congratulate all of your team that it is a very well organized
Thank
seminar.
you, ma'am. Thank you so much. Um, your comments are really very much encouraging, and we look forward that we'll keep organizing such events and make best possible contribution to the research in the subject in sciences. And in fact, I must thank you all because it is the participation that makes the event a success. The whole credit of the success actually goes to the participants in the first place. So the chair for the session is joined. Welcome, Dr. Praveen Janda. I take this opportunity to introduce the audience to the chairperson for this talk, Dr. Praveen Jangda. Dr. Praveen Jangda is an associate professor in the Department of Chemistry of Kurukshetra University, Kurukshetra. He has various research publications to his credit in different national and international journals and different various conference proceedings. And he specializes in organic synthesis and purification techniques, structure elucidation, and molecular docking. His research interests include synthetic organic chemistry and developing new routes of new synthesis, development of novel synthetic methodologies using multi-component reactions, and incorporation of different bioactive pharmacores in single molecule using this approach. So I welcome you, sir. Kindly take over for the next talk by Dr. Jain Sindhu. I won't take much time now because it's already running quite late. Dr. Praveen Jangda, please. Dr. Praveen, are you there? I guess you had joined. You're not audible, sir. Some technical problem at the end of Praveen's.
Yes, I think
Yes.
my mic is working now.
Okay,
Thank you, Dr. Deepa Sharma, for your good introductions. And I am also thankful to the Dr. Gulab for contacting me. In front of the biodata is of Dr. Jain Sindhu. Actually, he's my very good friend. And I can say that he's also similar to my friends also, because we have done the work together in the research. He has done his PhD from the Delhi University after doing MSc from the MLN College, Yamuna. And at that time, I am as a teacher in the Khalsa College when he is doing the PhD, MSc in the Yamuna. I think I am audible?
Yes, audible, sir.
Okay, thank you. After pursuing PhD, he has gone for the postdoc and he is done on the biosensors at the IIT Ropar. And he has published more than 45 research publications in the reputed journal. 4 students are working with him for the PhD, and the thesis awarded to the MSc students are 2, Ms. Neha and Ms. Komal. The highest impact factor of the Dr. Jayant is 9.02, which he has published in the articles. He is an organic synthetic chemist, and now he is working on the bioinformatics, particularly the molecular docking and the dynamics using the Desmonds. He is also very expert in the computational work of the Gaussians, 09 and 16 version of the Gaussians. At present, I think he is working on the biosensors, and his talk is on the topic of this ESIPT in the biofield. Now I invite Dr. Jain for his valuable talk. Dr. Jain, please.
Thank you very much, sir, for this warm welcome and for such nice words. Actually, what I have like achieved so far. You have contributed a lot in my early stage of my career, and I learned a lot of research techniques in your guidance. So thank you very much for your nice words.
No, it is your hard work, Dr. Jayant. Okay.
May I have permission to start my presentation?
Yes, you can start, please.
Thank you, sir. My screen is visible to all.
Yes, sir,
Yes,
visible.
visible, Dr. Jayant, visible. It is visible.
So today I'm going to discuss one of the established mechanism in fluorescence spectroscopy, that is ESIPT, excited state intramolecular proton transfer. And this particular mechanism has been extensively utilized in different type of medical diagnostic techniques, and many of the techniques have been developed using this particular mechanism. So first of all, I want to make you aware about what fluorescence is and how we can use fluorescence techniques in like different type of medical diagnostic. You might have heard about different like imaging agents which we can use to differentiate between normal cells and malignant cells or apoptotic cells, and we can also differentiate different type of tumors in our body. And we have a lot of example that are based upon fluorescence imaging techniques. One is we have in our routine, that is we have some chips that are supposed to use for some medical diagnostic test, like we can use for corona test, and we can have a pregnancy test also using some lab on chips. So fluorescence techniques have been evolved to a great extent and have a great role in medical diagnostic, particularly in if we want to achieve some test in short term, then we can use lab on chip anywhere that is handy to use, and that is cheaper also. So with the evolution of such techniques, the medical diagnostic have been progressed to a great extent. So today we'll start with one of the established mechanism, that is intramolecular proton transfer, that particularly happen in excited state. So intramolecular proton transfer is a photo-excited process that particularly results in those molecules which have a capability to show intramolecular hydrogen bonding interaction. And this particular mechanism has been proposed for the first time by Weller in 1950 when he proposed this particular mechanism for salicylic acids. So the ESIPT mechanism includes the transfer of a hydroxyl proton to an oxygen or nitrogen acceptor within a hydrogen bonded system. These are different type of representative compounds which can show this particular mechanism. So we have like hydroxybenzimidazole, hydroxybenzoxazole, hydroxybenzothiazoles, quinolines, benzophenone, flavone, anthraquinone, benzotriazole, and salicylbene anilines. So this is the mechanism, how this ESIPT mechanism takes place. When we have a framework, in that framework we have the probability of a hydrogen bond, which can say there is a possibility of some proton transfer within the molecule can exist in enol and keto form simultaneously at a and sequence of time. In that case, when we excite our molecule that is in enolic form, and enolic form is more stable in ground state, it will absorb energy and it get excited to enolic excited state. Then in excited state, there is a proton transfer that will take place, and it result into keto excited state. The keto excited state is of lower energy in the excited state, but the keto state is of higher energy. So when the proton transfer take place from enolic to keto form, there is a like shift in wavelength that we can observe. So in particularly we can get two different type of signals. One is from enolic form and other is from keto form, and these the ratio of these two signals depend upon different type of factors like solvent interactions, or we can say isotopic effect is also responsible for such type of proton transfer. So by using this particular mechanism and by utilizing the presence of proton in our fluorescent probe, we can recognize different type of analytes, or we can screen different type of biological processes also. This is a general representation of common processes that particularly takes place when ESIPT happen. So whenever we have a dye and we link some of the probe with the oxygen atom, we can replace our proton, the proton present in the form of enolic OH. We can replace that proton with some analyte, and if we excite this particular dye, we'll get only the enolic form emission. But when we cleave this particular bond by using some of the like analytes, we can use copper for ester bond cleavage. We can cleave C triple bond containing compounds by palladium. So there are different type of analytes which we can use for the cleavage of this particular OP bond. And by cleaving this particular bond, we can regain the OH proton in our dye. And once we regain our proton in our dye, then there is a possibility of proton transfer, and this proton transfer leads to keto emission that will be of like higher wavelength. So by like linking some of the analyte with our dye, and by breaking that oxygen analyte bond by using some different type of analytes, we can sense a range of like metal ions and different oxidant by using this particular mechanism. This is one of the first case in which ESIPT has been explored, and you can see we have three different forms of hydroxyflavons. There is a possibility of keto-enol tautomerism from this OS to this carbonyl group, and due to this keto-enol tautomerism, see we can see here below in this mechanism. First of all, this proton get transferred from this OS to this carbonyl oxygen, and on complete transfer of this proton, a new form, that is K form, that is formed in the excited state, and we will get emission from particularly from this K form. We can see the absorption spectra of this particular molecule. If we consider only this part of absorption spectra, the emission spectra is like a mirror image of absorption spectra, and we can see a band in the starting of this emission spectra of this particular molecule. When we record the emission spectra of this molecule at 77 K, means at low temperature, when we record, there is a possibility of only enolic emission, because the enolic form is more stable in ground state, and when it excites to some excited static enolic form, for proton transfer it have to cross this energy barrier, and when temperature is low, the energy required for crossing this barrier cannot be provided, and that's why we will get emission from this enolic form only. But when we increase the temperature, when we record the fluorescence at 273 Kelvin, we'll observe a large Stokes shift in the fluorescence emission, and the fluorescence is observed in green region. Initially, that was in violet region, that can be shifted to green fluorescence region. So this is the case, means how by varying the temperature of our studies, by recording the fluorescence emission at low temperature, we can differentiate between keto and enolic form. And when we have two different type of emission in our compound, by generally by restricting to only one emissive peak, we can recognize different type of fluorescent probes. Today, I am going to represent some of the good examples which have been reported in literature, and they have been extensively used in like biosensing techniques for different type of analytes like proton sensing, and different type of metal ions, oxidants, and then we'll show you some of the work from our lab in which we have developed some sensor for hypervolemic condition and for differentiating normal and apoptotic cell. So this is one of the spirobenzopyran molecule that has been developed for the sensing of proton. As we all know that proton is the smallest but most important cationic species that is involved in various physiological and pathological processes. So if there is a damage to transporter that maintain pH homeostasis is known to result in cellular dysfunction. So there is a strong need to develop some reliable sensors which can monitor even small changes in pH of a biological system. So if we have an insight, means how the pH of a system is varying, then we can recognize different type of biological processes. In this particular case, the spirobenzopyran, that is closed system, when there is a proton, when it it comes in the vicinity of a proton system, or when it comes in a like acidic environment, the closed system get open and it result into the formation of myrocyanin system. So when there is a myrocyanin system, see, there is a proton transfer probability between this OH and this nitrogen, and due to this probability, the possibility of excited state intramolecular proton transfer occur in this molecule, and we'll observe a significant enhancement in the fluorescence intensity from 557 to 686. So around like more than 100 nanometer shift that has been observed when there is a pH change in the medium. Similarly, one of the ESIPT probe that is based upon Rodal dyes has been Rodal fused HBT fluorophores that has been proposed, and that has also been used for lysosome sensings. As we know that lysosomes are membrane bound organelles with internal acidic environment, and the morpholin group serves as a targeting unit for these lysosomes. So one of the group has developed a ESIPT based probes, and they have attached one morpholin to the probe so that this morpholin can bind to the lysosome, and we can monitor the acidic environment, or we can monitor the change in the pH of the environment where lysosome present. So when there is a change in pH, then ring opening takes place, and due to ring opening, the initial form in which there is a possibility of proton transfer get converted into a keto form in which there is no proton. So when there is a change in environment, there is a change in acidic environment, the proton transfer probability get also diminished, and that's why we'll get a emission. We'll get a emission that is due to ESIPT off situation. So we by using these type of different strategies, we can develop some novel frameworks, and we can bind some different type of targeting group which can bind to membrane bound organelles, and by binding to these membrane bound organelles, we can detect different type of internal environments. Similarly, one of the group has prepared one of the sensor that is having the probability of ESIPT, and they have monitored slight changes in the environment, or slight changes in the concentration of magnesium plus 2. Magnesium plus 2 is an abundant element that plays a vital role in number of cellular processes, including cellular proliferation, a range of enzymatic reaction, cell death, DNA stabilization, and signal transduction. So Callan and co-worker designed a simple ESIPT probe for ratiometric detection of Mg plus 2 that works at 2 millimolar concentration in THF water system. Once there is a bond formation between this Mg plus 2 and the probe, the emission due to this ketoenol tautomerism get decreased, and we can differentiate between the normal emission that is due to ESIPT and the emission that we had observed after addition of Mg plus 2. Similarly, one zinc plus 2 probe has been developed, and by using this particular probe, see, after adding zinc plus 2, the possibility of ESIPT get reduced. So there is an large increase in enol fluorescence emission, which coincide with the appearance of a strong NIR keto emission peak at 7, 1, 10 when zinc was added to the system. This is one of the interesting molecule, since benzothiazole, hydroxybenzothiazole have been extensively utilized in the development of ESIPT based probes. And by linking, see, what I have discussed in our previous slide, that if you incorporate some of the group that can be cleaved by using some of the metal ion or some oxidant, then you can regain the ESIPT process. Similar is the case here. Fluorescent probe has been developed by linking hydroxybenzothiazole with piconyl ester moiety. And when this particular molecule is treated with copper plus 2, there is a cleavage of this ester bond, and again the ESIPT process get revived. So the limit detection of this particular sensor is 16.1 nanometer, and that have been used in detection of copper plus 2 in living cell also. This is one more example in which ether linkage has been cleaved by using such ESIPT probes, in which cobalt plus 2 is detected by cleaving a bond between this carbon and oxygen. This is one of the probe that has been developed for Fe plus 3, and in that case, when Fe plus 3 bind with the nitrogen atoms, then the process, that is ESIPT process, get reduced, and a turn-off response is observed. Because whenever iron is not present in this system, two type of emission were observed. One is for keto and other is for enol. But in presence of iron, only one emission has been observed, and that is due to turn-off response of ESIPT process. This is one more example in which, like vinyl ethers can be hydrolyzed selectively by using mercury, and different type of fluorescent probes have been developed on hydroxybenzothiazoles. We can see a visible change also when Hg plus 2 has been added. There is a shift in fluorescence intensity from blue to green. Similarly, a sensor has been developed for palladium plus 2 that is also based upon same mechanism, in which the oxygen carbon bond has been cleaved by using palladium plus 2, and there is a increase in fluorescence intensity up to keto emission that has been observed. And the limit of detection for palladium in this particular case is 87 nanomolar. There is one more example that is also based upon hydroxybenzothiazoles. In that case, two different type of analytes have been attached with the fluorophores, and by using like superoxide sensing can be done, because superoxide cleave this bond and it revived the ESIPT process in both the cases. Again, one more moiety that is based upon benzothiazole, but in that case we have amino benzothiazole. We can have both type of fluorophores when we have to develop some ESIPT based probe. We can also develop hydroxy benzothiazoles, or we can have amino benzothiazoles. The main concept is all about hydrogen bonding between two closely situated electron donating and electron withdrawing group. When we have a framework, in that we have two groups which are closely situated, one is acting as an electron donor and other is acting as an electron acceptor. In that case, we can transfer proton from one of the position of the molecule to another position, and we can have ESIPT response in that case. In this case, peroxynitrate sensing can be achieved by treating this benzothiazole based compound with peroxynitrate, and there is a visible change that has been observed when this peroxynitrate were treated with this dual probe. As we know that peroxynitrate is known for its destructive properties, causing irreversible damage to a range of biological molecules such as lipid, proteins, and DNA. So there is a strong need to develop such probes so that neurodegenerative diseases, cancer, inflammation can be sensed in their early stages. Sulfite sensing can also achieve by using hydroxy benzothiazole substituted compounds. In that case, also we can see by using sulfite. Sulfur dioxide is a toxic pollutant that is produced by a variety of industrial processes, and endogenously generated SO2 act as a signaling molecule in several physiological processes like cardiovascular disease, cancer, and many more. So Song and co-worker had developed a levulinite ester functionalized HBT fluorescent probes for the ratiometric detection of sulfur dioxide molecules. And they have detected sulfur dioxide from 0 to 1000 millimolar concentration in cellular system. Similarly, hypochlorite can also be sensed by using hydroxybenzothiazole based systems. In that case, this dithiolane functionalized HBT molecules have been developed, and once these dithiolane functionalized HBT molecules are treated with hypochlorite solution, they again regenerate the carbonyl group, and due to regeneration of this carbonyl group, there is a process of ESIPT then can be possible. In this particular case, there are two ESIPT process which are probably possible in this particular case. One proton transfer can take place from this OH from this nitrogen of the benzothiazole moiety, and the other proton transfer can be possible from this OH to the carbonyl group of this phenyl ring. So when we treat this particular probe with hypochlorite solution, then carbonyl get regained, and we can regenerate the carbonyl group, and we can have different type of emission. So this is one of our research article that we have published in NJC, that is disaggregation induced ESIPT, a novel approach towards development of sensor and for hyperglycemic condition. First of all, we have rationally designed our fluorescent probes depending upon the binding cavity of HSA protein. We have doped our designed molecule in the binding cavity of HSA protein, and it has been observed that our molecule overlaps the co-crystallized ligand that is present in the protein. Then we decided to extend some of the structural features of the molecule by adding one OH group that can behave as a probe for ESIPT, and we have extended one of the aromatic rings so that the lambda max for fluorescence can also be enhanced, and we can have signal like in our NIR range, and that can have application in biological system. So for that we have synthesized two molecules. One of the molecule is having a OH group that is closely situated with this nitrogen and have a probability of proton transfer. And the other molecule which we have synthesized that lacks this OH molecule. The purpose of synthesis of these two molecules is to differentiate either the response is due to the presence of ESIPT process or that is a normal fluorescence response. So firstly, we have explored the most probable confirmation by using DFT studies, and it has been observed that antiform is less stable. Antiform, when this OH group is towards oxygen atom, is less stable than synform when this OH group lies towards nitrogen atom. So by establishing the conformational stability of this particular molecule, we have utilized this molecule and we have explored its photophysical properties. While evaluating the photophysical properties of both these probes with OH and without OH, we had observed that in case of OH containing probe, we had observed two types of emission. One is from enolic form and other is from keto form. And in addition to these two emission, when we record the fluorescence spectra of our compound in water, we had observed one more peak at higher wavelength, that is due to aggregation induced emission enhancement. And that peak has also been observed in case of the second probe that lacks OH group. So the main structural feature of both these groups are, in both the cases, the presence of aggregation induced emission is present, and we had explored the phenomena in detail by varying the amount of water in THF system. And it has been observed when we increase the water content, there is a decrease in fluorescence emission, and around 90% of water, a shift in fluorescence wavelength has been observed due to formation of aggregate. And we can see a visual change in like this particular solution is having 80% water, and this particular solution is having 95% water. So above 90% there is formation of aggregation, and this aggregation is responsible for this large shift in fluorescence wavelength. Then we have evaluated the sensing application of our molecule by using different type of analytes like metal ions, some proteins, and some of the biological amines. And proteins we have selected depending upon their alpha helix content. Like we have selected two of the proteins, one is HSA and second is BSA. HSA is having more alpha helix content in comparison to BSA, though the structural feature of both these proteins are similar, and we can choose in some condition. If we don't have HSA, we can choose BSA because the structural features are slight same in both the cases. And we had observed that when we treat our molecule with these different analytes, two different peaks are observed, that is at around lower wavelength. When we add HSA or BSA protein, we had observed two different peaks at lower wavelength. This particular peak is due to aggregation induced emission, but when we shift our molecule, when we add HSA to it, we get a emission that is at lower wavelength. And in previous slide, we can see this particular emission that is around 450 nanometer, that is due to keto emission. So now it has been inferred that whenever HSA protein has been added to our system, the molecules which are in aggregation form, they get disaggregated, and they get attached to the cavity of the protein, because the binding between protein and binding between protein and our developed probe have been established in the starting of the study. Then we have titrated our ligand with different concentration of HSA protein. Once we have established the binding of HSA protein with our probe, then a conclusion has been made that the binding is due to the presence of alpha helix content in our protein, because those proteins we have also explored some of the simple proteins like simple tyramine and all that. In that we don't have alpha helix content, means these are beta sheeted protein. So the main difference between an alpha helix and beta sheeted protein is that alpha helix proteins are having some hydrophobic cavity, and when our molecule get disaggregated, it gets stabilized into that hydrophobic cavity in keto form. That's why we observed a signal due to keto emission at lower wavelength when we add some protein molecule in our fluorescent probe. Then we observe the time response also, and then we glycate our HSA protein by using different concentration of glucose. And as we know, when glycation occurs, the alpha helix content of the protein get distorted because there is a unwinding of the protein that usually takes place, and due to that unwinding, the alpha helix content get reduced. So if when we treat our glycated human serum albumin with our fluorescent probe, it has been observed that the peak that is observed due to addition of HSA and the peak that is observed due to addition of glycated protein have a significant difference. And the difference is due to distortion of alpha helix content in case of glycated serum albumin. So from all these studies, we have concluded that our our fluorescent probe have a probability to differentiate between glycated and simple human serum albumin. And by different type of binding studies with our HSA protein and our fluorescent probe, it has been observed that our fluorescent probes get bind to the alpha helix of the protein, and also with the tyrosine and tryptophan residue of the protein. So overall, we have concluded that if we have the ability, by using this fluorescent probe, we have the ability to differentiate between simple HSA protein and glycated HSA protein. And we know that in diabetic condition, when the content of sugar get increased in our blood, it generally binds with this serum protein. And serum protein is one of the it is one of the only protein which have the ability of transportation of molecule to different part of body. And once glycation occurs, the alpha helix content of the protein get reduced, and due to that, the different type of physiological processes which are going on in our biological system also get hampered. So by developing such fluorescent probe that is based upon desegregation induced ESIPT process, we can differentiate between glycated and non-glycated human serum albumin. And this is the primary step in diagnostic when we have to diagnose diabetic condition in a patient. If the content of glycated protein is more in a patient, then we can differentiate that particular content by using some fluorescent probe, or by developing lab on chips by using such fluorescent probes. There is one more article which we have published for the differentiation of undifferentiated, differentiated and apoptotic cells, and that is also based upon ESIPT. After successfully evaluating the hyperglycemic condition by using a naphthyl fuse system, then we have reduced the size of ring in this particular system. We have reduced one of the ring, and we have synthesized two molecules. In one molecule we have OH at the ortho position, and in second molecule we have OH at the para position, to evaluate the effect of ESIPT in this case. So in this case also, two forms are possible, syn and anti form, and syn form is more stable. As the stability of this syn form has been established by using different potential energy scans, which are done by using Gaussian software and by using different DFT methods. And it has been observed that the molecule that is more stable when it is in syn form, it means the molecule is planar and the energy is at its lowest potential energy surface. And when we rotate this molecule, when we have a dihedral angle scan, it has been observed that when the molecule get rotated by 10 degree angle, the energy get increased. And when it changes into anti form, if we compare the energy of syn and anti form, then syn form is more stable. Not even the ground state, but in excited state also, we have explored two excited state, S1 and S2. In both these cases, the ground state of this syn form is more stable than anti form. Then we have also evaluated the oscillator strength. Oscillator strength is generally a term that can be used to evaluate the absorption capacity of a molecule. Either the molecule is showing a good fluorescence response or not at a particular conformation. By evaluating the oscillator strength, we can explore the possibility of fluorescence or intensity of fluorescence in a molecule at different conformation. In that case also, it has been observed that when the oscillator strength has been calculated in first excited state, the oscillator strength was found to be greater when the molecule is in syn form, and the oscillator strength was found to be greater when the molecule is in syn form in second excited state also. So by by using this particular idea, it has been assumed that there is a hydrogen bonding between this nitrogen and this OH atom, and this hydrogen bonding restrict this carbon-carbon rotation at room temperature. And due to the restriction of this rotation, the non-radiative process get reduced, and we probably get the emission peak due to radiative transitions only. Then first of all, we have evaluated the different photophysical properties of the molecule in different solvents that I am not showing here. But the main thing is we have evaluated the effect of glycerol concentration on the fluorescence intensity of our molecule. We had observed that when we increase the concentration of glycerol, a significant increase in fluorescence intensity has been observed. Then we have also explored the radiative decay. Like we have also reduced the decay time also when the glycerol concentration has been increased in our solution. And it has been observed that upon increasing the glycerol concentration from 0 to 99, there is a increase in decay time. And by evaluating this decay time and by evaluating the quantum yield at different concentration of glycerol water, it has been observed that on increasing the viscosity of the solution, the radiative rate constant get increased and the non-radiative rate constant get decreased. And this is the main cause of fluorescence enhancement when we change the concentration of glycerol from 0 to 100% solution. And by initially establishing the effect of viscosity on the fluorescence intensity, then we have evaluated our molecule in cellular imaging, and it has been observed that undifferentiated, differentiated and apoptotic cell can be resolved by using our fluorescent probes depending upon viscosity, because apoptotic cells are more viscous than undifferentiated cells. Then we have also evaluated the cell viability at different concentration so that we can observe the cytotoxic effect of our dual probe in cellular system. And it has been observed that our molecule different cells exhibit around like 60% of 80% of viability up to a concentration of 100 microgram per ml. So by using such type of dual fluorescent probes, we can sense the viscosity of cellular system, and we can diagnose different type of pathological condition like cancer in our body, because by using these probes we can differentiate normal and apoptotic cell. And similarly, we can also attach our molecule with some targets which can bind with mitochondria or with lysosomes. So ESIPT is one of the best technique which has been extensively utilized in past few years for the development of fluorescent probes, particularly in medical diagnostic. And I hope it can prove a boom to organic chemists and also medicinal chemists who are particularly working in the development of novel fluorescent sensors for biosensing, so that the cost of medical diagnostic can be significantly reduced. Thank you very much.
Thank you, Dr. Jain. Thank you very much for the nice talk on the ESIPT. And Dr. Jain has discussed the ESIPT regarding the acid sensors, oxidizing sensors, and hypoglycemic sensors. How can we determine the hypoglycemic agents also, or some programmed cell death using the apoptosis and viscosity sensor. In the acid, here discussed that we can sense the protons, magnesium, zinc, copper, iron, mercury, palladium, like Lewis acid sensors. Or in the oxidizing agents, such as the superoxide sensing, peroxynitrite sensing, also there. Sulfide sensing is there also. These are the oxidizing agents which can generate the free radicals in our body, and if their sense are known or we can sense them, then it can be utilized, or this method can be utilized in the biofield to remove the aging process or remove the cancer process also there. So such nice talk has been produced by the Dr. Jain. Thank you very much, Dr. Jain. Now house is open for the questions for the audience. If audience, they can ask the questions or their problems to the Dr. Jain. I think I read some about the one person have raised your hand, that is the Chandra Prakash. Mr. Chandra Prakash have raised his hand. Okay, now the mic is handed over to Dr. Deepa Sharma.
Well, thank you, sir. Thank you so much, Praveen, sir, for sparing time to chair over the talk. And thank you so much, Jan, sir. I appreciate your profound research work. And it is opening many new avenues. And I hope the researchers present with us will get many new ideas from your research work. And anyone who wants to collaborate with Dr. Jayant, kindly share your email ID in the message box, please, so that anyone can contact you for the ideas of collaboration. Okay, thank you, sir. Thank you, Praveen, sir. Thank you, Jayant,
Thank you
sir.
very much, ma'am, for giving me this opportunity to present my research work in this international conference. And thank you, Dr. Parveen, for sharing this session for me. Such a nice experience I have with you. Thank you very much, sir.
Okay, Dr. Jain, I have also shared a teacher and every time for you. No
Okay,
problem. We can have this time. Okay, thank you. Thank you to the college and the authorities of the college for organizing such nice conference and inviting us such good lectures. I'm also thankful to Dr. Gulab who contacted me regarding the chair session, particularly for this event. Thank you to everybody.
Well, carrying forward, the next talk will be chaired over by Dr. Sushil Kumar Pandey. I guess Sushil sir has joined. Sir, are you there?
Yes, I am present.
Welcome, sir. Welcome to our conference. And thank you so much for sparing time. I would like to introduce the audience to the chair of this talk, Dr. Sushil Kumar Pandey. He has done his PhD from Indian Institute of Chemical Technology, Hyderabad, and he's right now assistant professor. And he has professional experience of 16 years after PhD. He has served in Joseph Fourier University, France, as postdoc research associate, Albany Molecular Research, Hyderabad, India, as senior research scientist, Jubilant Chemsys Limited, Noida, as assistant director. Various rewards and recognition to his credit. He has got Best Team Award 2021 for a project. He has been awarded as the Best Scientific Manager Award from Jubilant Chemsys in 2014. Special Leadership Award from Jubilant Chemsys in 2015 for exemplary contribution. Best Team Award in 2010 for outstanding performance. So, very vast industrial as well as research as well as academic experience he has. So I welcome you, sir. We are privileged to have you with us. A number of publications to his credit and many conference proceedings, invited lectures he has delivered. So over to you, sir. Kindly take over the session, please.
Thank you so much, ma'am. Thank you so much. Thanks a lot for the nice introduction. So this is my privilege to be part of this conference. And thank you so much to the organizing committee, especially Suresh and Dr. Gulab. So like I will take over and will introduce Dr. Manpreet Rawat. I think he has joined already, probably.
Yes, sir. He is in, sir. He is in.
Very good. Thank
Hello.
you so much.
Yeah,
Dr.
I'm here.
Manmeet
Thank you.
Rawat.
Yes, Dr.
Thank
Manmeet
you very
Rawat.
much.
Yes.
Mhm.
So I would like to introduce Dr. Manmeet Rawat. So he has a vast experience in the research field and is working as a assistant professor at Penn State University. And he did his education from Haryana, and master's degree he did from sorry, just 1 second. Masters he finished from Maharishi Dayanand University, Haryana, then did his PhD from ICMR, Dwarka, Delhi. Then did his postdoc from the University of Minnesota School of Medicine. He has been the member of different societies and educational boards, has got many awards to his credit. Few of them would like to inform here is in 2020 he got the Biomedical Research Excellence Award, 2019 Research of Distinction, many certificate of recognitions he has got in many years, and then Gordon Research Award in 2012 to name few of these. Also, he is editorial board members of many journals and reviewer of many of the scientific journals. He has many publication, close to 4 dozen of publication, and he is regular in invited lectures. He has been presenting continuously in scientific participations. He is taking care of many of these things. So I would like to invite Dr. Manmeet Rawat to start with his presentation, and we would like to welcome him here.
Before sir start, with the permission of the chair, I would like to extend a request, please.
Yes, please.
Sir, as we are running behind the schedule, so I would request you to kindly take care that the speaker takes 10 minutes less than the allotted. Okay?
Sure.
Thank you.
Thank you very much, Dr. Pandey, and thank you to the college, the organizing committee and everybody to invite me. And don't worry, I'll try to take as minimum time possible because it's already midnight at my place, and it's already around 1 a.m. in the night time. So don't worry. I myself want to take as
thank you so much
as possible. Thank you.
Yes,
and
please.
Can I proceed?
Yes, please.
Thank you very much. Can you see my screen?
Yes.
Okay, Ji. Very good morning to all the listeners. And actually, this is such a new experience, having all the conferences online, and we are used to with that regular way of talking. But I'll try that things go smooth and everybody will understand. And I hope most of my audience today is from college background. Please correct me if I'm wrong. Is that right, Dr. Pandey?
Right. Yes, you are right. Yeah.
Okay, so I'll try to keep it as simple as I can. Although it's a hardcore molecular biology, drug discovery type of work. So just for the information for the audience, the young guns, young students from the from colleges. I'm pretty sure most of you are from chemistry background.
If the chair permits, may I add something?
Yeah, sure.
Yes, please.
Sir, I would like to inform you that most of the participants, they are researchers and faculty members of chemistry background.
Yeah,
thank you so much. That's good to know.
Yeah, thank you.
So might be all of you, you are people from hardcore chemistry background. So might be you are thinking why I'm here, or why Dr. Gulab Singh invited me to this. So let me make it clear. Chemistry as such like have different dimensions, but what common people know that chemist, they people from chemistry background, researcher from chemistry background, what does they do? So for
Sorry,
it,
Dr. Manmeet, your voice is actually very slow, so just be a little bit close. Thank you.
is it good now?
Yes, very good. It is good. Great.
Thank you. Okay, so I was saying like most of you like for the common people actually, if we go around and we ask common people what is chemistry and what is it all about, what chemists do, what researcher from chemistry background do, so most of the time we will see okay they are making some type of drugs or medicines or like products which can benefit human body. So here we go. So what microRNA is? Why I'm here with the chemistry conference? So most of you know, as you said, most of the audience is like chemistry background researchers. So there are like different ways of like the drugs we discover or we invent, how they work. And in that category, there is a new type of thing where we talk about epigenetic control of any disease. What does that mean? Epigenetic control means the control of any function or any process in our body by our own genomic components. So that is why the new hope for the complicated disease like different cancers or different type of things, the new hope is epigenetic control. And here comes a very minute, very small molecule to which we call microRNA. What are these by microRNAs? So, like as the new slide says, why microRNAs? So if you see nowadays, like after human genome sequencing, we found some like really interesting things around which were not earlier like not discovered or which were not talked about. So usually we used to see like our genome, human genome is consist of like different genes and some silent genes and this and that. But recently we came to know like a lot of genome don't have like uh, like people don't have much information about most of our sequenced genome, and it is a talk of like hardly uh, 20 years, something years. So what is these microRNAs? These are part of our genome. They are like really, really small molecules. How small? Hardly 19 to 20 base pair. And what is these bases? Not pair, actually base. So let me tell for the common audience, our genome, human genome is consist of different genes. And these genes, they like make mRNAs which turn into protein, which make like protein, and these proteins are base of our life. Like and these proteins are everywhere. Like our whole body is made up of these different proteins actually, and they are the one who control the function in our body when there is a diseased condition. Like for example, there is some cancer. So what happens, like some gene out of like so many genes, like some particular gene can go off actually. Off means like it can either increase the production of protein or decrease the production of protein, and ultimately turn into a like diseased condition. So these like genes, they make mRNAs like RNA. These are another molecule in our body, in our cells, and they are like DNA. I'm pretty sure most of you are aware about these words, DNAs. RNA, I'm pretty sure some of you are aware about it. So these RNAs, what they do, they get information from our gene and then they make protein. And what are these proteins? These proteins, as I said, they are base of our life. They are like different, whatever like you call, we talk about different, like how our cell function and all. So these proteins are the real thing which turn like all the activities. And these proteins actually. So there are like 2-3 level of like controlling of these proteins. Sometime like cells or our genomic system, they turn down the production of the proteins. So in normal condition, for example, one protein is made to, we have a like most of my research is around GI tract infection, actually gut infection, our intestinal infections. So like for example, some gene is responsible for controlling the intake. We know we eat every day, right? Every time we eat like minimum 3-4 times, or like all the time we keep eating actually. Where it goes to our intestine. From our intestine it goes to our body. So there is a check at that level. So for example, we have a skin outside our body. That skin is a protective layer outside, and it's a multi layer. So if we like scratch it, it still remain there. Some of it can come out, but inside our like gut there is a single layer of cells which protect from like whatever we are eating. For example, like when we eat so many other things, like we have hundreds and thousands of like different microbes which stay inside our gut. So what happens, like sometime this one single layer of the cells, it get defected because of like there are like some particular proteins. We will talk about them later on. So like what happened to these proteins, their production goes down. How it goes down? There are some particular microRNAs which we studied, which we published. They control the function of these proteins. Where they control, they control it at the level of mRNA formation, which I said comes from like DNA. So let's talk about a brief history of microRNA. So like microRNAs are not very old molecules. They are old, like as old as we are, or human being, or any cell, or any living thing on this earth. But they got discovered in 1990s, and that time nobody took it seriously. So people used to think they are like really small molecules and what they will do. So it was hard to understand. So more and more we are studying our human genome. We are coming to know that okay, these are like really important things, and they like contribute some important function. So as I said, what are these microRNAs? We talked about it. They are like small non-coding molecules. Why I'm saying non-coding molecules, I'm pretty sure most of you are new about these terminologies. So let me tell, as I said, mRNA comes from DNA. DNA is like what? Like our genes, right? From our gene, we need information. We keep listening. I'm pretty sure this much all of you are aware about that our genes, they have the information coded in them for every function in our body. Like whatever function is going on in our body, it got regulated by genes actually. And how these genes regulate them? They create some molecules. And those molecules, some of them are coded for some particular proteins, some are not coded for anything. So earlier we used to think those non-coding molecules are good for nothing. But as more and more like science is progressing, things are getting better, we are understanding more and more about our body. So we came to know that these non-coding molecules are also there for some function. So this microRNA is one of them. And microRNA biogenesis, I will skip it. I'm pretty sure this is more for biology background people. They can understand it more. So I'll skip it. So here, I'm not sure if you can see my cursor, but you can see there is a big strand. What are they? These are our RNAs actually, and we can see there is one long red color of the threads or whatever you want to say it, and then you can see a pinkish element or pinkish thread is attached to it. So this pinkish color thread is microRNA, and this reddish color, the bigger thread is mRNA. What is mRNA? mRNA is a molecule which comes from, as I said, which get information from our gene or our DNA. So like this microRNA is attaching to it and degrading it, and that way protein production goes down and it creates problem in our cells. So why we are talking about what they will do? So like as I said, there are some common examples actually. This is one slide which talks about like there are different type of disease which are like somewhere associated with these microRNA dysregulation, and some of them are like you can see different type of cancers are there, different type of like heart diseases are there, some nervous system related problems are there, and some inherited disease which comes from like parents to child, they are there. So these microRNAs are responsible for them. One is like progressive hearing loss. So what happens like as kids grow, as the child, those who are having this condition from they get from their parents, when they like grow more and more, they'll keep there is a progressive hearing loss. So why it happens? It happens because of one microRNA, which is microRNA 96. These are like these words like 96, 184. These are like common way of nomenclature for these microRNAs. So then there is another condition where skeletal and growth defect happens, and it is because of another microRNA whose number is 17. So microRNA 17. And so there is a long list of that. I'll skip. As you said, there is a crunching of like we are short of time. So okay, let's talk about what we are doing. So and why like I'm talking so much about these microRNA and what we will do. So usually what we can do like as we create like being a chemistry people, as all of you, like most of you or some of you, those who do research in drug discovery, so what we do, like even we do drug discovery, being a biochemistry by biotechnology background person. So what we do, we try to like synthesize some molecules. What these molecules usually, what is their function? Like whatever drug we are taking, for example, and any drug, like name it, and paracetamol we are taking, what they do, they go inside our body. These molecules somehow like they go to their target, and they either like stop their function, they like inhibit their function, and sometimes they are like very specific for some particular molecules, like which are like responsible for those particular diseases, or like like uh, what we can say, the unnatural condition. So we recently published one paper, and what is that paper? Here it is. This paper is got published in one of the like number ranked number one journal of gastroenterology. Gastroenterology, let me tell a little bit. Gastroenterology is about like our gut. So whatever gastroenterologists are the doctors who does gut related treat gut related issues, and gastroenterology is branch of like medicine which deals with gut related problems. And most of our research work is revolve around it. So this is like here we can see, you can see my cursor. Hello.
It's visible, sir.
Okay, so okay, so in here we can see what I'm talking about. So like sorry, I cannot see it. Okay, now I can see. So here we can see this is like we took a mouse model actually. So most of the studies we usually do on mouse actually. So in first picture you can see there is a normal control mouse, actually healthy mouse. And then in second picture, you can see what happens. We call it colitis, ulcerative colitis, Crohn disease, or inflammatory bowel disease. These are like three words you will keep, like you'll listen in my, like further in my talk. So in here we can see, like in second picture, healthy gut, human gut or animal gut, looks like the first picture. And then in disease condition, what happens, these are, we call them microvilli. These are part of our healthy gut actually. They are the thing which takes nutrients and everything from our food, and then it goes to our, inside our body, and then we survive. Or so in this condition here we can see there is no villi actually. So what happens, inflammation create ulcers. These ulcers like destroy all these natural structures, and it create problem for our body. So here we try to understand why it happens, and like our background tells us, whatever work we did in our past studies, that tells us that cytokines, these are some of the things which create issues actually in our body. So we try to understand and we try to block it. So let's go. So what is impact of our work? So it will help to strategize to block the effect of one cytokine, which is responsible for ulcerative colitis condition. And this study will give us some hope that we can create some antisense molecule of these particular microRNA, which we identified. So they are like drugs actually. They are like medicine. So we are trying to create antisense, means these molecules, they are also they are also microRNA. What they'll do, they'll go and they'll bind to these microRNA, they will stop them, and then they will ultimately protect our cells from this particular cytokine. So that was like cover story. So let us understand our gut a little bit. I don't know how much like you guys are interested in it, but yeah, I'll try to explain. So this is our inside of our human gut, intestine. It looks like this. These are different cells, and this is our lumen, where everything remains, like including hundreds and thousands of microbe with our food. So it remains here actually, and these cells, they allow them to go inside. What they allow, it depends upon them. They know, like our body is like best machine on this earth. So our body knows what is good for our to take in, what not. So here, as I was talking, so what these microRNAs do, like this is our nucleus. They produce it somewhere here, then they go to the the cell, and then so these are the different cells. As I was talking, this is like one monolayer. So these are different cells. I just did magnification, and it's a pictorial diagram, so don't worry, it's not the real picture. What this is almost a real picture, but this one is like just a pictorial diagram to understand it. So these cells, they are attached to each other through some barriers actually. So what these microRNA I was talking about, what that microRNA usually do, they disrupt these barriers or these gates, what we can say. And what will happen then? Here we can see like these are disturbed one, and this opening is like they create this opening. What will happen after that? These microbes and everything, they are coming inside our body. So our whole efforts are like to stop it. So here is some data. I will not go much in like detail of it. So as I said, these gates, they are consist of like different proteins, and we studied one of the protein which is like major part of these barriers, and we call it occluding. So we try to look like what is happening this with this occludin and what this microRNA is doing here. So the first data which you can see on the screen is from cell model. So what we do, we usually study all these things at three levels. One in like cells, which we grow them in inside the lab on a petri plates. We grow them and then we do study on them. Second model is animal model, means mice or rabbit or different. Ours is mice. So what we do, like we try to study everything on uh, these mice and try to understand what what is happening with our these animals. And third is human tissue or human organoids, which we call, and we call them mini organs also. So don't worry, I know I'm giving so many new words to you, but leave it and do not think much about it. Just think this is like the study we did with uh, cell model actually, and we try to see what is happening at different levels. As I talked about occluding protein, so we see that in disease condition, occluding protein production goes down. It started we started this from 0 hour to 72 hours, and here we can see you can see these are the bands. Actually we isolate these proteins, we run them on different gels, and we try to see the production level. So you can see a gradual decrease, and when it is happening when our body, our gut is in diseased condition. This is one cytokine. I don't know how many of you are aware about these words, but I'm pretty sure most of you know these words, cytokines. So what happens in disease condition, the cytokine level goes up and then it create inflammation. So what is happening, particularly we know pinpoint now that occludin is one protein which goes down. So we try to understand it in different conditions. We give this iron usually in our body, it comes naturally, but here we are giving the cells this iron beta directly. So we get it from different sources, and then we directly treat these cells with this one. So here you can see again production going down, and then we try to study it at different level. Here we can see at mRNA level, as I said, occludin is protein synthesized our body. How it comes from as I said, gene create mRNA create protein. So yeah, then we try to see if their junctions are intact, and this data shows that now in the presence of island beta, the barriers are not intact, and the mRNA level is going down, and we measure transepithelial resistance. What is this resistance? This is this is the thing which tells us whether cells are healthy or not, or their barriers are broken or not. So when resistance goes down, it means uh, the, the cells are getting in bad position. Here we can see at 72 level, this one is going down, and then we try to see it with we choose different size of molecules and we try to pass them through these cells, and here we can see this flux is going down. So that shows that cells are not in good condition. And then we try to understand the level of expression again in with the help of, as I said, we identified this micro RNA which is responsible for it. So what we did, we created one anti micro RNA molecule, and here we can see, as I said, that when iron beta is there, this microRNA level is too high. In control condition, in healthy condition, it is really low. So we created these anti-sense molecules against microRNA. As I said, in iron beta presence, microRNA level goes up. So we blocked it with anti-microRNA. molecule, and here we can see that microRNA level is going down. What it is doing when microRNA level is going down, the occluding protein, which I like said earlier, in normal condition it is very high. It make our cells healthy. So when we give anti-island beta, it the cells get into bad like situation, and then when we treat them with this antisense molecule, we can see the production of occludin is coming up. And these are different ways of looking into it. This is at protein level, that was at mRNA level, and like this is again we try to see if as I said, we use this flux, like we try to pass some molecules through these intact cells, and in control condition you can see it's like very low, and in island beta condition flux is high. It means gates are open, and we try to like treat them with antisense molecule, and here we can see again that it comes to almost like a normal condition. So let's move to the another one. Then we try to study it in animal model, as I said, and this is animal data where we can see that in island beta presence, the flux we talked about like in the last slide is like really high. It means the gates are open between these two cells, and uh, our like cell animal is not in a good condition. Animal gut is in a bad condition, and we check this microRNA also, this particular microRNA which we think regulate this function, and in island beta condition it is again very high. Then we try to treat them, and before that we try to see again that occludin. So we do it at different level, actually same thing we are doing at different level, and yeah, that's all what we are doing here. And then again, as I said, we created these antisense molecules, and we try to use them and check what is happening in like in animals. So here you can see in control condition, this microRNA which we talked about, the microRNA. No, sorry, I think I missed the exact name. So microRNA 200C is that particular microRNA. So, okay, sorry. So here we, okay, I'm sorry, I think it's something bad. So here we can see like when we treated them with antisense, we can see it goes almost like control condition, whether it is that particular occludin protein level, whether it is microRNA level, whether it is the flux which we use to understand, again as I said, like to understand what is happening. Then we try to see like this is as I said earlier, like this is animal data. These are the pictures from the actual animal, and here we can see in control condition, this is how our gut or animal gut looks like. In disease condition, it like turn into a mess. There is no clear, we call them microvilli. There is no these type of clear structures here. So what we did, we gave that antisense of microRNA, which we identified, and you can see the condition is almost recovered, like somewhere like a healthy one, not like 100%, but almost 80-90%. And this is the disease index actually. So in diseased condition, so how we know whether this animal is sick or not, I'm pretty sure some of you are aware about these things, because being a chemistry person, you guys are like I'm pretty sure you are testing your compounds somewhere with animals, and so what we do, we check body weight time to time for these mice, like these animals, what is happening with their body weight. So when we created this disease condition, the body weight is going down as time progresses. So like we looked up to 7 days. So as time progresses, the body weight goes down, it means the animal is sick. So when we treated these animals with the antisense molecule, we can see it recovered actually. Again, it's almost 70-80%, not 100%, but yeah, it recovered and animal started feeling good. Then we checked, as I said, at third level. Third level is human tissue. So we got human tissues from patients. Like as my workplace is a, like as I said, it's a health science center or school of medicine or college of medicine. So we like regularly get these human, like diseased human or like patients in our hospital. So we got these tissues from them, and we try to see the level of microRNA 200C, which was our interest, and we created those antisense against this microRNA. So in disease condition, you can see the production is very high. And same, like we created another model. To them we call organoids. I'm pretty sure it's a new word for most of you. Organoids are like, we call them mini organs. So what we do, we try to create these organs in Petri plates or in lab. So these are the organoids where we studied them, and they also show the same, almost same condition. This is another data where we try to go into 3D modeling. Pretty sure some of you are already doing 3D modeling for different molecules. So here we did 3D modeling for microRNA and mRNA. What is it like? If I'm just reminding you, what is this mRNA? This is like mRNA comes from DNA, and try to this mRNA, they code the information for protein. So which protein we are talking about? The occluding protein. So this is the mRNA molecule for having information for that occluding protein. So what this microRNA does in blue strand is our microRNA. So it bind to it and try to degrade it, try to stop it, try to it work in different ways. So here we can see, we try to see, this is one way of looking. Relative luciferase activity means we check the activity of these mRNAs, and in when there is only microRNA and there is no when this microRNA is there, we can see production is going down. And when we deleted these sites where this microRNA binds, the effect goes away, and we can see the production is coming to the normal level. So that's all. To conclude, I just want to make it simple. So our data shows that IL-1 beta or these cytokines or in disease condition, microRNA 200C expression goes up, and we try to create antisense of these. And these antisense, they are helping us to recover, particularly in mouse model. In humans, these things are still need to be tested, but in animal model they are working very beautifully, and they are doing a really good job. That's all, and I would like to take as many questions as you have. I know it was a very fast explanation of everything, but yeah, you can go ahead and
Thank you so much, Dr. Manmeet. It was nice topic, and the science that you people are doing really great. So if there are any questions, we can take up, Dr. Deepa.
Yes, please. Anyone from the audience, any questions? Welcome. I guess that's it.
Actually, I would like, it will be great actually. I'm pretty sure most of you were feeling lost in this presentation because it is out of the regular area. But yeah, if you have any question about anything, do not worry. Every question is a good question. So just ask if you can. I'll be happy to answer. Ji, Dr. Pandey.
Yes, sorry. So actually, how this our artificial intelligence is going to help in this drug discovery or like micro RNA type. Any idea on that?
Actually, I think you are mixing these two talks. One was AI based talk. I think Dr. Anis Abrol, I think, supposed to do that. Okay, so definitely, artificial intelligence is such a beautiful thing. It can help in almost every possible field of our life. So let me tell a little bit. So our human genome is such a complex thing. We have so much work to do. So to understand human genome accurately, we need better tools. And these artificial intelligence programs can, first level, they can try to help us to make things easy. Because for a human brain, it is almost impossible actually. Till now, we are using different software. They are also capable of this information from
Dr. Manmeet, your voice is slowly. It is not coming good.
Okay, now it is coming good
Yes,
or
yes,
like
it is good. It is good.
sorry, my bad. So I'm saying like artificial intelligence is such a smart tool which can help us to understand our human genome more
Yes.
accurately. And
Yes. True.
ultimately, when we talk about create some therapy or some drug or some treatment or some medicine or some epigenetic control mechanism, we need
True.
to understand so many different aspects of
Yes.
uh,
Yes.
interactions. And artificial intelligence is such a good thing. It is developing slowly, and even we try to use artificial intelligence to understand these micro RNAs or to identify these micro RNAs. Actually,
Yes.
till
Yes.
now, our human genome is uh, hardly half decoded, even less than that.
Yes, it is. It is. I think it is not that explored yet, and we are just
Right.
generating the data on that basically, right?
Right. So it is such a huge data. It is almost impossible for human brain to process it.
True.
And
True.
these whatever softwares we developed, they are also
Mhm.
not they are trying their best but still feeling lost. So I think AI is the
Yeah. So actually, we were working on these things, and like I'm working with a company. They are working on this data generation and artificial intelligence, and they are trying to utilize that. So I thought this is like related to the structure and the binding. Probably that studies will be coming up. So I thought asking to you.
Oh, definitely. Like in disease condition, when we, okay, for example, like our human body is, as I said, one of the most complex machine it is. So in a disease condition, like when we look, there can be thousands of different ways, like
True.
our cells are getting in bad shape. So to understand that, for example, we talked about this one microRNA, right? Before
Yeah.
that, we took years, like it took, I think, 5 to 6 years to screen these microRNAs actually.
Mhm.
So we started from a big number actually, like 20, 30 microRNAs. Then we,
True.
come to like 4 or 5. And then we
True.
ultimately it took us like 4 or 5 years to come to that one microRNA.
Mhm.
AI can help in that. AI can, predict better. It can
Yes.
make things
Yes.
better, and it can try to yeah.
Thank you, Dr. Manmeet. Thank you so much. It was a nice talk, and like I know those who are working on drug discovery probably will be understanding better. But the molecules are required to test in that, so it is good to correlate these two things, and like it is helpful for all of us to understand that. Thank you so
Yeah,
much.
thank you so much, and I am pretty sure after this talk, more of you are going into the internet and looking for these words, and yeah,
Yes.
these are new ways actually, future of
Yeah.
medicines actually. Epigenetic control is future of
Yes.
uh,
True. Very true.
medicine. So
Yeah.
thank you.
Thank you so much, Dr. Manmeet Rath. I think Dr. Deepa, we can go through the next ones. Or
Well, thank you, sir. Thank you so much. Thank you, Manmeet sir, for sharing your research work with us. And I'm sure it will be beneficial for all of the participants, and they will get leads for their own research. And now, going ahead with the conference session, I would like to invite Dr. Krishna Kumar from Deenbandhu Choturam University of Science and Technology to chair the next talk, please. Before he takes over, I would like to introduce you to Dr. Krishna Kumar. Dr. Krishna Kumar received his MSc and PhD in 2007 and 2013 in chemistry from the Department of Chemistry, Kurukshetra University, Kurukshetra. Dr. Krishna Kumar is presently working as Assistant Professor in the Department of Chemistry of Deenbandhu Choturam University of Science and Technology, Murthal in Sonipat. More than 10 years PG teaching experience also he has. He is a reviewer of several ISI indexed international journals. Moreover, he is one of organizing secretary and member of technical committees in various international conferences and seminars. In addition, he is an active reviewer of many international journals and conference proceedings also. He is an active member of many societies and committees. His area of research lies in nanoferrites, doped metal oxides, and nanocomposites. He is supervising PhD students, MSc students in the Deenbandhu Choturam University for the thesis work. And to promote science, Dr. Krishna works as organizing member of 2 science conclaves for school students also. So I welcome you, sir, and I would request you to kindly take over as the chair. The next speaker in line is Dr. Badri Prasad. Dr. Krishna Kumar will introduce you all to the speaker, please.
Good morning, ma'am.
Morning, sir. How are you, sir?
Fine, fine, ma'am. Hello, Dr. Badri. How are you? Dr. Badri, your mic is muted.
Yeah, sorry, sir. Yeah, thank you,
Okay.
sir. Yeah, I'm good.
Okay. So, a very good morning to one and all present here. I am Dr. Krishna from Department of Chemistry, DCRAS Murthal. I welcome you all in the technical session 3 of the International Conference on New Trends in Chemical Research 2022. So today we have with us Dr. Badri Prasad, who is currently working as a research associate in the FRUC research group, Department of Chemical Engineering and Biotechnology, University of Cambridge, UK. Dr. Badri received his master's from Department of Chemistry, Kurukshetra University, Kurukshetra, in 2011. He did his PhD degree from Department of Chemistry in synthetic organic chemistry from the same department. Dr. Badri have published number of research paper and delivered a number of invited talks in many international national conferences. So with these words, I would like to invite Dr. Badri to deliver his lecture. And the topic of his talk is Influenza Virus Inhibitor Multivalent Polymeric System. Dr. Badri, please.
Yeah. Yeah, thank you, sir, for nice introduction. And I don't know if I send you the wrong short biography, because 2 months ago I joined the University of Harvard as a research fellow. I have finished my studies at the University of Cambridge. So, and now we are going to talk about influenza virus inhibition by multivalent system, and most of the work about influenza virus inhibition is done at the Freie University of Berlin, and some of the work is done at the University of Cambridge. And influenza, you all are familiar. Maybe you don't know the word influenza. Maybe you know the word flu, which is very common in all over the world and around like in the winter time. And if we talk about the flu cycle, yeah, sorry sir, I forgot to ask, can you see my screen?
Yes, Dr. Badri, you can see.
Okay, thank you sir. So influenza virus cycle or flu cycle start with the entrance of virus to the respiratory tract system, and this occur as the same like as we see in the current situation with the coronavirus or SARS-CoV-2 virus. And the flu cycles occurs in like different stages, and it affect our lungs, our respiratory system, and it like start with the like entrance to the respiratory system on the day first. And in the day first, usually we don't see any symptoms. If you are infected with the virus on the first day, if you are in contact with some person who is already infected, or if you maybe touch any surfaces which is infected by the virus, and after that if you touch your nose, eyes, or your mouth, you can be infected by the virus. But on the first day, and sometimes within like 48 hours, you don't see any symptom. But after that, you you start to develop the symptom, and the major symptom for the influenza are like fever, body shivering, headache, body pain, and runny or congested nose. And after day 5, like symptoms become even severe, and up to day like 7, and in some cases like up to day 10. And it depends like person to person, like how long you get the symptom and like how severe are the symptom. And sometimes like some people with strong immune system, they just recover in a week, but normally the cycle lasts for like 10 to 14 days to recover from the virus. And if we talk about the structure of the virus, and the structure of the influenza virus is quite similar to the SARS-CoV-2. This is also a corona virus. It has spikes on its surface, and but unlike SARS-CoV-2, it has like 2 spikes protein on its surface. One is called hemagglutinin, and one like second one is neuraminidase. And these 2 spikes protein helps the virus to interact with cell and infect the cell. So in like throughout our talk, we will say like hemagglutinin as a HA and neuraminidase and as a NA. Like basically these 2 are the 2 like important hand of influenza virus. So influenza uses this HA protein to attach on the sialic acid on the cell surface. So this HA protein has very nice affinity towards sialic acid. So it uses is HA protein to attach on the sialic acid and enter into the cell. And after entering
Yes,
into the cell,
Chairman sir, if you comment, I would like to interrupt, please. Sir, I would like to mention that your presentation is not visible on the screen. The last slide by the last speaker is visible only.
okay, 1 second, let me exit. And
Deepa ma'am, it is visible to me.
Sir,
now
is it influenza virus and its surface proteins rule?
Yes, ma'am.
yes,
Yes, sir. It is visible. So, some participant just told me.
Okay.
Okay.
Thank you, ma'am. Okay, thank you, ma'am. So as I mentioned, influenza has 2 important spike protein. One is HA, which helps the virus to enter into the cell, and second is NA. Because after infecting the cell, when virus comes out of the cell, then this HA protein is still attached on the sialic acid. Then in this case, this NA protein help to cleave the binding of HA to sialic acid, so that the virus can release from the cell and can infect the second cell. So it is the similar, like if we see, if like one hand, if I try to simplify it, like influenza uses one hand to open the gate and to enter into the cell, and it uses its second hand to exit from the cell. So what happen if we block the second hand? Like when it exits from the cell, this first hand is still attached on the gate. Like if it's stuck in the gate, like in the door like this, if we hold the second hand, then it can't exit from the cell. If it can't exit from the cell, it can't enter into the second cell, and we can inhibit its spread. So most of the drugs which are available in the market actually are neuraminidase inhibitors. It means they hold the second hand of the influenza virus. It means influenza virus can enter into the cell, but it can't exit from the cell. So most of the drugs, and the most common drug which are recommended by doctors is like Tamiflu, and which is like the chemical name for this drug is oseltamivir, and other drugs are zanamivir and peramivir. And as I mentioned, the like not most, like all of the drugs are neuraminidase inhibitors. So but scientists also thought about to inhibit the HA protein. So why not like we just inhibit the entry of the virus to the cell? Because if we inhibit the HA protein, then HA protein can't attach on the sialic acid and it can't enter into the cell. If it can't enter into the cell, it can't separate. So scientists thought about that, and what they did, they tried to synthesize the same sialic acid which is present on the cell surface. They try to synthesize and they try to isolate from the natural source. So what scientists did or researchers did, they use this sialic acid, which is the same which is present on the cell surface. They try to inhibit the entry of the virus to the cell, but what they found, the interaction of this single sialic acid with virus is quite weak as compared to the interaction of virus with the cell sialic acid, because on the cell the sialic acid present in like very huge amount, and they have very strong binding as compared to the single sialic acid. And if we compare the quantity, this like in 10 raised to power minus 13 molar, and the single sialic acid in 10 raised to power minus 3 molar. It means there is a difference of like 10 to the power 10 molar in terms of activity. But researchers try to like shorten this gap, and what they did, they try to synthesize different types of multivalent inhibitor. It means they try to synthesize the same type of architectures with sialic acid which is present on the cell. It means they put the same sialic acid not in the single form but in multivalent form. They put on different polymer or different nanoparticle. And this multivalent system, why we call it multivalent? Because we attach the same copy of sialic acid in multiple manner in like different like surfaces or different core. So this idea is basically inspired by like nature or by like man-made multivalent system. If you see these natural system, they are multivalent system. For example, if we see the DNA, these two DNA strands are in like form double helix when they form like multivalent hydrogen bonding. If you just try to attach two base pair, like just single two base pair, then you will not form double helix. Even up to 5 or 10 base pair don't form a double helix. They just stay in like single helix in solution. To form double helix, we should have multivalent interaction, even more than 10 multivalent system, multivalent interaction. So that's how we know the multivalent interaction are very important. Even in some like synthetic or some man-made system, the multivalent interaction are very strong. If we see, you see the example of this man-made Velcro. If you try to attach this, just this tiny single hook with tiny thread, you will not able to hold. But if you have like hundreds of tiny like hooks and hundreds of tiny thread, you have like strong binding, which we can see in our daily life. So by taking inspiration from these, researchers developed several multivalent systems, and what they found, if they attach these multivalent sialic acid on some nanoparticle, on some dendrimer, or on some polymer, they can inhibit the virus very efficiently. And with this finding, they found that these linear polymers are much stronger binder than the dendrimer one, because the linear one are much more flexible, and in one go they can attach to several different proteins on the influenza surface. Like this is the temp image of influenza, and this like one spike protein like corona of the influenza is around 13.5 nanometer, and it has 3 sialic acid binding site. If we have rigid system like some rigid nanoparticle or some dendrimer, we can bind just like 2 or 3 site in one go. But if we have some flexible, we can bind even like with the same molecular weight, with the same functionalization, and with the same structure. Just one is linear and one is dendrimer. We can bind even like more than 4 or 5 because of the flexibility. So we try to take advantages of this flexible core and this multivalent system, and we explored some multivalent system to inhibit the growth of influenza virus. And in our case, instead of using just sialic acid, we are using zanamivir as well, which is like neuraminidase inhibitor. It means we want to inhibit the growth of virus or spreading of virus, like by inhibiting the attachment of virus to the cell, and also by inhibiting the release of the virus to the cell. It means if we inhibit the HA protein, we can inhibit the attachment of virus to the cell. If we inhibit the NA protein, we can inhibit its release or exit from the cell. So we try to use these two molecules, sialic acid as a natural like drug or natural molecule, and zanamivir is a synthetic drug which can synthesize in lab, and we can synthesize a bivalent system with dual mechanism of action. So to synthesize this system, we first modify our drugs, or we synthesize our drugs in 12 steps by starting from sialic acid. And as you can see in zanamivir, there are like many functional group like acid, amide, guanidine, and hydroxyl group. And if we see the interaction of neuraminidase with zanamivir, we found that this like C7OH group, this OH group doesn't participate in binding. Like if we see the binding of zanamivir with this NA protein, this OH doesn't participate. All other functional group are participating in binding. It means we can modify this hydroxyl group to create some other functionality, and we can retain the activity of this drug. If we functionalize other hydroxy group or a carboxylic or amide group, we lose the activity of the drug. So that's why we functionalize this hydroxyl group with alkyne, and in the similar way, we functionalize this silyl lactose, which is the derivative of sialic acid and even more active than the like this sialic acid. This is silyl lactose. We functionalize both with alkyne to functionalize our polymer, and which have azide, and by using click chemistry, we synthesize several different derivatives, and 3 representative molecules are shown here. So we synthesize bifunctional, which contain sialic acid and zanamivir. It means we can inhibit hemagglutinin and neuraminidase both, and we, for the comparison, we synthesize their monomeric analog as well, like monofunctional, just sialic acid and zanamivir. And by doing their activity against different strain of virus like X31 Panama and Bahrain strain, like we have for now like different strain of coronavirus like Omicron, Delta, and Omega, and different strains. So in influenza virus also we have different strains. So we tested our compounds against different virus, and if we see the IC50 value of this compound, we found that drugs has like in the range of like 3000 to 5000 nanometer, but our compound has value only like in like low nanomolar range, even we can say in picomolar range. So from here, and also when we study the effect of these two molecules, these are also like much more active than zanamivir, but in comparison to this bifunctional, they are less active. And even if we check the physical mixture of that, like if we mix both these compound and test against the virus, then again these both two are less active than their covalent mixture. And by studying different physical mixture, we found that the physical mixture also show the activity similar to like this zanamivir compound, because this active like interaction of this zanamivir with neuraminidase is much more stronger than the interaction of sialic acid with the hemagglutinin or HA protein. That's why this always overcome the activity as compared to sialic acid. If we take the mixture of these, that is shown the activity similar to just single zanamivir only. And then we tested our compound and checked by different techniques, and we checked like cryo-TEM images as well. Without the virus, our like polymeric system, they form some planars, like 2D planar system as like shown by P here, and some condensed, like some aggregated structure like shown by C here. But in the presence of virus, they all form just aggregates. And what we concluded here, like in absence of virus, these intramolecular carbohydrate interactions are much more stronger, and they form some 2D nanostructures. But when we treat them with virus, then this interaction of the sugar with viral protein is much stronger than their intramolecular interaction. So in this case, their intramolecular interactions break and they form interaction with viral spike protein, as we can see by cryo-TEM image. And also when we check the confocal images of our particles with influenza virus, we can see without any virus. And here we labeled the virus with green fluorescent dye. So without virus, we can't see any fluorescent, but when we treated our cell with virus, we can see all over the cell there are a lot of virus. And when we treat these cells with commercial drug, then we can see this influenza virus like reduction throughout the cells. Like we can see a lot of like more than 50% reduction. It means we can inhibit the virus like more than like 50% concentration by using different amount of zanamivir. But if we treat this infected cell with our compound, we can inhibit more than 90% of virus. Here you can see we can see just few small dot of virus which are labeled with green dye. And here we concluded that like our this bivalent system, which are functionalized with sialic acid and zanamivir, is much more active than commercial available drug and their physical mixture and their monovalent analogs. And so in the second project, we tried to explore the other nanoparticle where we can even see the much stronger binding of our like our polymeric system. So in second case, what we did, we choose some virus-like nanoparticle because virus has spikes, and we created some virus-like nanoparticle. And from the literature, we found that these virus-like nanoparticle, even without any functionalization, they like are responsible for the inhibition of virus. And the reason behind that, because the influenza virus spikes from the nanoparticle, they insert like into each other, and in that case these nanoparticle don't allow the influenza to interact with cell. But the binding in that case is not very strong, and we just see like just 20% of the virus inhibition. Like 80% of the virus is still available to infect the cell. So in that case, what we thought to functionalize these nanoparticles with our synthesized molecules. So we use both molecules like the sialic acid and zanamivir containing polymer. We functionalize with these virus-like nanoparticles with these molecules, and again we tested against different strain of virus, again like some X31 Panama and some PR strain, and we also compared the spiky nanoparticle with smooth nanoparticle when there is no spike. So these are just nanoparticle without any spikes, and these are virus-like nanoparticle VLNP. And here we found that these like spiky nanoparticle, these one are much more active than this smooth nanoparticle. And even if we compare these bifunctional inhibitor with their monofunctional analogs, and we can see these are monofunctional. This is just sialic acid functionalized, and this is zanamivir functionalized, and in this is bifunctional. These bifunctional inhibitors are much more active than their monofunctional, and in all three tested strains. And again we check the confocal image of our nanoparticle, and in this case we labeled our nanoparticle with red fluorescent dye. And here in case of like control, you can see a lot of viruses are there, and when we use virus like nanoparticle just with sialic acid, we see inhibition of virus, but still there are a lot of virus. It means we don't see like 100% inhibition. But when we check and the virus in presence of nanoparticle which are functionalized with both sialic acid and zanamivir, we see like more than 90% of the inhibition. As we can see, there is hardly any virus like red color seen in confocal microscopy. And again we from here we came to know that like morphology of the nanoparticle is much more important in the inhibition of influenza virus. So we collaborated with some research group where they synthesized some nanoball type structure, which is like concave type structure, and they functionalize these nanoball with RBC membrane. And actually these RBC membranes are natural source of sialic acid. So we collaborated them, and we suggested to check the inhibition of influenza virus with these like nanostructure which are functionalized with RBC membrane, had sialic acid, and when they checked the inhibition of the influenza virus, they found some sort of inhibition because they have sialic acid, and sialic acid can block the activity of hemagglutinin. And then we synthesized some zanamivir containing compound so that we can insert in that, and again we can have bifunctional inhibitor. So here they call them like heteromultivalent system, because there again like we can call also bifunctional inhibitor or heteromultivalent system. And in this case again this zanamivir can inhibit the Na protein and sialic acid which are naturally present on RBC membrane, can inhibit HA activity. And from the cryo-TEM or TEM images, we found that these like carbon nanoballs are quite symmetrical, and they have the size of around 150 nanometer, and which is quite suitable to inhibit influenza virus, because influenza virus are roughly in the range of 100 nanometer, and these 100 nanometer can nicely fit into these concave structure of these nanoparticle. And from the theoretical calculations and some computational study, we found that if we use like some inhibitor which are spherical or convex shape as compared to concave, they have less contact area, and even they have less steric hindrance. But in case of like this nanoball or concave, we have more contact area and more steric hindrance. It means we can just use less concentration of concave as compared to convex. And from the studies here we can see again we tested against the same three strain, Varian, Panama and PR, and here we can see heteromultivalent system are much more active than their homovalent system. So this is like some brief discussion about my work, which I did in Freie University Berlin and Cambridge University UK. And in short, or in conclusion, what we found, like these bifunctional inhibitors are better than the monofunctional inhibitor, and even their covalent functionalization, like if we covalently functionalize just two like different inhibitor on same polymeric chain, they are much more active than the physical mixture. If we have just one on one polymer chain and second on second chain, if we mix both and treat, then this is not good. The covalent part is much better, because if we have just physical mixture, then Zanamivir compound don't allow the sialic acid compound to come closer to the influenza virus, because they have much more stronger binding with the influenza virus spike protein. And with the other nanoparticle like spiky versus smooth, we found like spiky nanoparticle are much better even without any functionalization. They show some sort of inhibition, whereas smooth nanoparticle without any functionalization, they don't show any inhibition. And also the concave nanoparticles are better than their convex counterpart, because of the much higher steric hindrance and much more contact area with the virus. And with this all, I thank you all for listening, and my team from Freie University Berlin, where I worked for like more than 2 years to synthesize these compounds. And all the activity, like biological activity, are done by Stadmuller and me. And I was guided by Dr. Sumati Bhatia in the Haag group there in Berlin. Yeah, thank you all for listening. And yeah, let me know if you have any question regarding this.
Okay, thank you, Dr. Badri. Thank you very much, Dr.
Yeah.
Badri Prasad, for very informative and exhaustive talk. Now the session is open for discussion. So, questions from audience. Anybody who want to interact with Dr. Badri?
And okay, if not now, you can contact me later as well. Like I have mentioned my email address here, and you are always welcome. If you have any question regarding these, like influenza virus inhibitor, or anything about the synthesis of these. Hello.
Hello, Dr. Badri. This is Manjit Nehra from Central University of Haryana.
Hello, sir. Thank you.
Please, Manjit.
So I'm curious to ask one little question that you mentioned somewhere, that some binding of this nanoparticle with this COVID spike, that I'm interested to know that which type of how you evaluate the binding, like with some molecular docking or some other
No, I think we can't use molecular docking for this complete virus. Maybe for simple, like single protein, if they are not attached to virus, we can use like molecular docking. In some cases, like we tested, like before testing these compound, we just tested like these HA protein and NA protein, like isolated with our compound, like with our Zanamivir and our sialic acid compound. We found like nice binding, and after that we like went for complete virus, but we didn't do any like molecular docking. We just see the inhibition by some biological assay in which we can just detect the binding, and even we can use some, I think, ITC also, but I didn't do that. And because the most of these studies are done by like my collaborator, and they have very nice facility where they just can use like cells with virus, and then they can use like confocal microscopy where they can see the inhibition, and then they count the number of virus in the infected cell and number of virus after treating the compound. And by calculating the number of virus, they can see like how much is the inhibition of the virus with these compounds.
Yeah. So I got it. Yeah. So these studies are going in lab, not by using this computational tool. So
No. These are done with cells and like virus, and even like we did some in vivo studies as well, but I didn't present here because of the like time limit. And in like case of in vivo results as well, we can see like very nice inhibition of influenza virus if we label our virus with some dye, and if we do some in vivo imaging, we can see the reduction of virus when we treat the mice with our compound.
yeah. Thank you. I got it. Also, Dr. Deepak send you hi. Dr. Deepak,
Okay.
he sent you hi, that say hi to Dr. Badri.
Okay. Thank you, Deepak. Yeah.
Okay. Thank you, Dr. Badri. Best of luck.
Yeah. Thank you, sir.
Okay, today's lecture was full of knowledge, and it gave us deep insight into the topic and also revealed very interesting facts. You have explained all the details and the basic concepts of the influenza virus. So we really grateful for the time and the efforts you took to share your knowledge with us. So that thank you very much, Dr. Badri Prasad, for sharing
Thank you.
your valuable
Thank you,
time.
sir. Thank you, sir, for the kind words and giving me the opportunity to present my work. Yeah.
Okay, now I would like to extend my thanks to the organizers of this two-day international conference for giving me this opportunity to chair this session. Thank you very much,
Thank
ma'am.
you, sir. Thank you, sir. Thank you so much. And my heartiest thanks on the behalf of the entire organizing team for sparing your time for us. We are heartily thankful. Thank you, Dr. Badri, for sharing your research work with us. Hopefully, many of the researchers will get good ideas from research work, and they will get good collaborators too, I guess. And Dr. Badri is always available for any kind of conversation through his mail. He has shared his email ID with you, so you are all free to contact him on any concerned topics. Thank you, sir. Thank you, Krishan, sir. Thank you, Dr. Badri.
Yeah, thank you, ma'am.
And with this technical session comes to an end, and we'll meet after a break of 15 minutes, please. Hello? So it's going to be noon, and we are back with the next technical session of our conference. We have amongst us to start this technical session 4 of the conference. The first speaker online will be Dr. Sushil Pandey, and Dr. Suresh Dua is here to chair the session. I welcome you both, sir. My heartiest welcome on behalf of all the organizers, on behalf of our college, on behalf of the Department of Higher Education. Dr. Suresh Dua is here to chair over the talk. Let me introduce the audience with Dr. Suresh Dua. Dr. Suresh Dua is working as an assistant professor in the Department of Chemistry, Kurukshetra University, Kurukshetra. Before joining Kurukshetra University, he has also served AIGHM PG College Rohtak as a lecturer for PG classes. He has a total of 16 years of research and 12 years of teaching experience for PG classes. He obtained his master degree from Maharishi Dayanand University, Rohtak in 2002. Then after he joined chemistry department, MD University Rohtak as JRF and received his PhD degree in 2009. His doctoral degree entitled Studies on Oxygen Heterocyclic Compounds includes the development of sustainable procedures for organic compounds of biological and medicinal importance. During his research, he also worked at Central Drug Research Institute, CDRI, Lucknow, a CSIR lab that is. Presently, his research is focused on green chemistry, catalysis, and medicinal chemistry. I welcome you, sir. Kindly take over as the chair.
Thanks, Dr. Deepa, and thanks to Dr. Suresh also for giving me this opportunity to be part of this international conference. Today we have with us Dr. Sushil Kumar Pandey as a speaker. He is an assistant director. As far as the education profile is concerned, Dr. Sushil Kumar did his PhD from Indian Institute of Chemical Technology, Hyderabad in 2005. He did his MSc and BSc board degree from Jaunpur University in 1995 and 1997. Dr. Sushil Kumar has a vast professional experience. He has served from 2005 to 2007 Joseph Fourier University, France, as a postdoctoral research associate. He worked from 2007 to 2009 at Albany Molecular Research, Hyderabad, as a senior research scientist. He also served, currently he is working with Jubilant Chemsys Noida and working there. He has recipient of so many rewards and recognitions, some of which are Dr. Sushil has been awarded Best Team Award 2021 for one of his projects, Best Scientific Manager Award from Jubilant Chemsys in 2014, Special Leadership Award from Jubilant Chemsys in 2015 for exemplary contribution, Best Team Award in 2010 for outstanding performance. Dr. Sushil Kumar is also having a vast research experience. He has delivered so many lectures in various international conferences and central universities and various other universities. Some of highlights are he successfully delivered collaborative medicinal chemistry program on kinase for the treatment of cancer and the compound as in clinical phase. He also worked on the synthesis of tubulectomins A as a part of his PhD thesis. Dr. Sushil Kumar have published many research papers in journals of national and international reputes, some of which highly impact journals are Tetrahedral Letter, Journal of Organic Chemistry in 2012, in 2007 also catalysis communication. So he is having a vast experience of research. So without losing much time, now I invite Dr. Sushil Kumar Pandey for his talk. Yes, Dr. Sushil, please.
Thank you so much, Dr. Suresh, for a nice introduction. I would like to thank the committee and Dr. Suresh and Dr. Gulab for giving me this opportunity to just highlight the things that we are working or where the pharma industry is going or which side it will be going and what will be the career probably. In that sense, I would like to talk a little bit. So thank you everyone for this opportunity. So if you permit, I can start my presentation.
Yes, sir. Please.
So do you see my slides?
Yes, it is visible.
Good, thank you. So actually, I took this topic, trends in pharma industry, CRO and its future. So according to the title of this conference, I took this and just wanted to highlight what are the things going on in the industry, some of the things which are coming up and what is going to happen in the next few 5 to 10 years probably. I just wanted to highlight some of these things that is going on currently. So I'll go to the first one. So this topic, whatever I am having, that will cover 4 things. So one is the continuous flow chemistry. Other one is AI, the artificial intelligence and drug discovery. Third one is the PROTACs. And the fourth is process versus CRO industry. So one by one, I'll go through. So this is continuous flow chemistry and its application in the production. So basically, continuous flow is one of the area where most of our production companies, they are working on this area very vigorously. And this is very important process which is going to come up. This will reduce a lot of things in the industry. So people are trying to work on this continuous flow chemistry. Previously, the reactors were used. So probably once this comes, and some of the productions are going on in the continuous flow one. So this is very important discovery that is coming up, and a lot of things are happening in this area. So everybody tries to reduce the time of the reaction, and if it becomes continuous flow, that means the batch process, whatever people were doing, it is going to reduce that timing. So batch process means when the batch starts, it will take a day or two to complete the reaction. But in the flow chemistry, it is just going to take some minutes of time to complete the mixing and get the reactions done very quickly. So some of the important points that we have here is the reactive components are just simply mixed in a tube, and the reaction, basically the reaction, when we talk about that reaction, timings are very less. It is femtoseconds time that reaction, many of these reactions take when the particular temperature and particular energy required is reached. So in like reactions when we are doing in the batch, so there we are giving more time for the reactants to react and the byproduct formation was happening. So with this flow chemistry or the continuous flow chemistry, when we are utilizing we will be having very less byproduct formation. Particular temperature, whatever we are talking about, it reaches in a very minute time, so that the byproduct formation or the intermediate formation, which is not stable, which going to react with other things, are very easily minimized. The reactions will not have, byproducts formation will be stopped. The reaction will go to the direction where it is required to go, basically. So this thing is coming up very nicely in the industry, and many of the companies, they are working on. Still, there is not very like everywhere it is not being utilized, but people are working on this, and it is going to be the future of the industry in like next 5 to 10 years time. This is going to be a very important discovery that people will be having. So simply one example we take, when we require reaction where the temperature is maybe 200 degrees centigrade, and the reaction completes in 5 seconds. there is no reaction vessel where we can do this type of chemistry in 5 seconds. But in flow chemistry, two things are mixing at the particular temperature, and then it is flowing to the other side, or the reaction is complete within 5 seconds. Two things are mixing up, and then it is going to the workup processes. So where we can just achieve that particular temperature even for 5 seconds for that particular reaction. This will happen in the continuous flow chemistry. And then some of the examples that we have. So these are the small scale examples that we were having. So the alcohol to aldehyde, if we are doing in the reaction, we know these aldehydes many times are not very stable. So that may form the byproducts. But in the continuous flow, if you see, 100% conversion is achieved, and very, very small time it takes for the reaction. Very quickly this performs the reaction, and the product is flown through these things, and it is passing quickly. There is no other time for the other things to happen. So as soon as this is reaction is done, it goes to the workup process where we have the water or any other thing to extract. So that will be done in very, very short period of time. This is one example. The other one is when we have this allylic alcohols, which are again not stable. Many byproducts formation happens. So here also, if we want to protect that alcohol as a methyl ether, so this reaction again is very short time reaction, just 20 minutes this reaction happens. But if we do this in a flask, this takes more time, and lot of byproducts formation are observed. So in the continuous flow process, these things can be avoided. Again, there are some other examples. If we take example of paracetamol, so the starting material, pyraminophenol, that is produced in industry by electrochemical process. So there again, it is a very simple process, and they have utilized the flow chemistry. Otherwise, if it is kept in the flask, or if it is reacting in other things, there are a lot of byproduct formation observed in that reaction. So by that, it is being controlled. So this flow chemistry or continuous flow is going to be the future of the industry. These are some of the reactors that we use. The right-hand side one is the small scale when we talk about the very small scale, or when we talk about the process development part. So these are the machines that are being used. In a day, I think 1,000 of reactions can be performed in these machines. It is like a simple HPLC machine that we are having. So we have to just put all the samples, all the reaction condition, 1,000 of condition you can standardize and then feed it in the computer. Everything automatically will have, and the next day you come, all the reactions data you will be having. So whatever the best method you want to take from there, you can take and utilize that for your standardization process. So standardization, once it is done, then slowly we built up the bigger scale. It goes to smaller, bigger scale. Like this will be the very small quantity, micro ml quantities will start with, and then we'll go for the liter or like the ml quantities, followed by will go for the continuous process. So the left hand side is the, like you can imagine how big the reactors look like. Here, this is the very simple reactor, which is the process flow reactor. So this is the area, just you can understand the big reactor, 5,000 liter and 10,000 liter reactors are replaced by this very simple flow chemistry machine, which will be producing more than what the 5,000 or 10,000 liter reactor is producing in a day. Because that reaction, it takes long time, but here continuously the reactants are flowing, mixing, and the product is forming very quickly. So this becomes continuous flow. There is no stop of the flow. One reaction, one time you finish it, then go for the workup, and all that is avoided. So here, this is going to be the continuously flowing reaction. So the reaction times are less, and your product conversions are good, purification will be avoided, and all that good things will be happening. Initially, we will need some process development in this. So that is what is happening at the moment in the industry. Many people are working on these type of things. So, but
Dr. Sushil,
yes.
sorry to interrupt. Probably slide is not transiting.
Okay.
Yeah. Sorry to interrupt.
No, it is now it is showing, or
yeah. Now.
Okay. So here the right hand side, as I discussed, the left hand side is the actual reactor actually. So this type of reactors now available. So you can see the figures right in the slide. Photos basically.
Yeah, it is visible now.
Okay. and then I'll go to the next topic, which is the artificial intelligence and drug discovery. So basically, this is again the topic which is coming up very nicely for the drug discovery. These are having like very good advantages over the like normal drug discovery that we do. Traditional versus the AI based drug discovery we talk about. So traditional is like we will have to decide which target we want to work. So targets are already known, and then we have to work on that, and we have to see whether it is druggable or not. So all that information has to be there in the traditional drug discovery. But if we talk about the AI based, so this is going to be the data driven, means all the algorithms and everything will be there, data will be there. It is not necessary that if something is undruggable previously will not be druggable. So we can find the targets very easily by the all the data that we are having in the machines will be having. They will be having all the data. You just feed that and what type of compounds it is required. So basically these things are again like at least 4 or 5 companies I know which are working on these data collection. So they are every day lakhs of data they are collecting, and they are feeding into the machines so that the machines can decide what target, which compound, what type of things are required for that processing. But again, this is again a very initial stage of the research that is going on, especially in the case of drug discovery. So still the data or the machine learnings are going on. So all the data that is getting deposited in that machines are being fed are going on, and it will be soon they will have most of these data available, and then accordingly, like whatever the compounds we want, whatever the target we want, accordingly the compounds, type of compounds, whatever the required things will be there, will be decided very quickly and very easily. So basically this application, what application will be having of the AI to drug discovery. So this is going to revolutionize the current time scale, basically the timelines, whatever, and the expenditure that we were having. So basically, if we go for the normal drug discovery or the traditional drug discovery, per molecules, whatever that comes into the market is around close to $500 million. But once we start utilizing the AI, that will be 1/3 or 1/4 of the cost that we are putting up. And the timelines initially, like we have to start with knowing nothing. And here at least we will be knowing some of the things where we are going to work. So the timelines also will be very short in these cases once the AI information comes up. This is another topic, the PROTAC. Actually, this is again a very important target that is going on in the industry. People are working proteolysis targeting camera. So basically, this is a bifunctional type of small molecule which will be having 2 active domains linkers, which will be like hijacking the cell ubiquitin proteasome systems and degrading the E3 ligase, like that will be target protein, and E3 ligase they will bind, and they will try to degrade the proteins that are responsible for these type of things. So this is another thing which is going on very recently. Like a lot of people are working, a lot of companies are working on this target PROTACs, and many of these are in the clinical phases. So these things, and one more important thing in this is like this is a catalytic process. So in this the quantity of the drug that we are giving will be very short, very less quantities will be given to the patients, and the side products will be minimized in the PROTAC cases. So the process of this actually, if you see this slide, so here in 2001 this concept came up, and 2008 we were having the first PROTAC molecule to target MDM2 E3 ligase. So that was done in 2010. Slowly some more things came up, and in 2019 we were having 2 PROTACs which were entered into the clinical trials. At this moment, I think at least 10 or 12 targets which are which are in the clinical trials, basically are molecules which are in the clinical trials are working going on. So this is again one of the most important topic that people are working on. So there are a lot of advantages if we talk about the protects. So drug resistance, whatever we were seeing previously in case of especially the tuberculosis and other drugs, so that will be avoided even in these cases. So a lot of other advantages that we are going to have with this protect. So that is again the one more important aspect that is going on in the industry basically, and it is going to be the future again in the drug discovery basically. One more thing I was having that actually this will help our researchers, those who are working for their masters as well as PhD. So the contract research and drug development versus the process development and manufacturing. So generics companies. So what are the opportunities that we are having? So basically we look into the process development companies are generic companies, Torrent Pharma, Cipla, Zydus Cadila, Arvindo, Sun Pharma, and Reddy's. These are the many, like there are many more, but I have taken some of the examples. So these people, mostly they are working on the generic version of the drugs. Some they are also producing new molecules, but mostly these are the generic ones. And if we talk about the CRO companies, so due to this COVID situations, I think India has got very good opportunity and does drug discovery in our like the CRO companies, contract research manufacturing companies. They are blooming like anything. So the things are going very well with these companies. If you talk about GVK, Biocon, and Jubilant that we are working on, Sai, we talk TCG Life Sciences. So these companies, now the number of hiring that is going on are much higher than the process manufacturing or the generic medicine manufacturing companies. If we talk about Jubilant, like we have almost hired 150 people in the last year for the PhD, and total almost 500 people we have hired. So if you compare the generic manufacturing companies as well as the CRO companies, so the difference that we see is going to be is very big. So almost 1,000 to 2,000 people we are hiring. This five or six companies only are hiring almost 2,000 people per year. And if we talk about the other generic companies, so not much hirings. So this is going to be the future again, the CRO discovery. So this I just wanted to highlight here for our people, those who are in the research or those who want to do the research. So this will be the future if they want to look into. So the CRO companies will be the future again in next 5 to 10 years. These are going to bloom in the country, in India especially. With these things, I would like to thank everyone. Thank you so much for this opportunity.
Now the lecture is open for discussion. We are still short of time, so we may entertain one or two query. Any query from participant? I think there is no query. So on behalf of organizers, I deliver my sincere thanks to Dr. Sushil Kumar for his wonderful talk. And this talk will really help the researchers to interact, to do their research work in according to the industries. So thanks Dr. Sushil for this wonderful talk, and thanks organizer for giving me this opportunity. Thanks Dr. Suresh and Dr. Deepa for giving me this opportunity. Okay, thank you.
Well, thank you, Dr. Suresh, for chairing over the talk, please. Thank you, Dr. Sushil, for presenting your research work with us, sharing your research work with us. And now moving ahead in the technical session, our next speaker online is Dr. Virender Singh, and I'll myself be chairing over the talk with kind request from the convener, Dr. Gulab sir. So, but as I'm also conducting, so let me introduce myself to you. I'm Dr. Deepa Sharma. I'm head of the Department of Physics in Shaheed Udham Singh Government College, appointed as assistant professor under the aegis of Department of Higher Education, Haryana. And my research work is in the field of computational nanophysics. As such, boundaries are fading. So it's chemistry as well as physics. You can say it's a kind of interdisciplinary work. My work is basically on single-walled carbon nanotubes. And I've done computational work on it. I have studied their their electronic spectroscopic properties using computational softwares like Material Studio पे मेरा basically काम रहा है. So moving forward, our speaker of the session is Dr. Virender Singh. Dr. Virender Singh has done his graduation and post-graduation from Kurukshetra University, Kurukshetra. He was gold medalist in MSc Chemistry. He qualified CSIR, JRF, and GATE with 97.32 percentile and completed his PhD from Central Drug Institute, Lucknow in 2011 in Synthetic, Organic, and Medicinal Chemistry. He got Best Thesis Award from CSIR CDRI, Lucknow. After PhD, he joined Central University of Punjab, Bhatinda in 2011 and worked as assistant professor for a period of 1 year. Thereafter, he moved to Department of Chemistry, NIT Jalandhar in 2012 and worked for 8 years as assistant professor. In 2020, he moved back to Central University of Punjab, Bhatinda as associate professor in chemistry. His area of research is synthetic, organic, and medicinal chemistry. Dr. Singh has published more than 62 research papers in peer-reviewed international journals and 2 book chapters. He has also participated in 32 national and international conferences. And at present, 2 research projects are ongoing and 3 research projects funded by SERB and CSIR have been completed under his supervision. Presently, 6 students are pursuing their PhD under his supervision and 4 have completed. So he has got several awards like Dr. M.M. Dhar Memorial Award, Professor S.M. Mukherjee Award for Excellence in Chemistry, Nandalal Telesara Memorial Award from Indian Council of Chemists, Young Scientist Award from Indian Science Congress. He was also selected for D.S. Kothari and prestigious CSIR Nehru Postdoc Fellowship. So we welcome you, sir, and the floor is all yours to share your research work, please.
Thank you, ma'am, for a nice introduction and kind words. At the outset, I would like to thank all the members of organizing committee and Government College, Matek Majri, Karnal. And I'm also thankful to Dr. Gulab Singh, who is also my classmate during graduation and post-graduation. Thank you all for giving me this opportunity to share my work with all of you. So, first I will want to check whether you can see my PPT and it is working. Ma'am, can you see it? Hello, am I audible, ma'am?
Yes, sir.
Hello, ma'am. Yeah, ma'am, my PPT is moving. Slides are moving.
Sir, we are not able to see your PPT.
Okay. There's some network issue. Hello.
Yes, sir, you are audible.
Yeah, ma'am, do you see my PPT?
No, sir, the screen is not shared.
I have shared, but I don't know. Okay, sir. Thank you, sir. So I want to check whether these slides are moving. Slides are moving. So now I'm going to second slide. So can you see? I think it's some network issue. Let me find there's some network issue with me here. Hello. Ma'am, can you see my PPT now? Okay. So the topic of my presentation is exploration of regioselectivity of Coumagian C for the synthesis of beta-carbolin based privileged scaffolds. Okay, now question arises, what is beta-carbolin? Beta-carbolin is basically a tricyclic framework that is made up of indole ring fused with the pyridine ring. You can see the A, B, and C ring. It is a tricyclic framework, and you can number it this way: 1, 2, 3, 4, 5, 6, 7, 8, and 9. You can see this is one position, and there are two nitrogen atoms. This is N2 nitrogen, and this is N9 nitrogen. Okay. And today we are going to discuss about the application of this natural product. This is Coumagian C. We call it 1,4-mile-9H beta-carbolin. And it is isolated from the root wood of Picrasma quassioids. Okay. So, sir, can you see my second slide? Okay, thank you, sir. So, why beta-carbolin based natural products are so important? The reason is the importance or significance associated with the alkaloids of beta-carbolin. Beta-carbolin alkaloids are ubiquitously present in nature, and they are found in plants, marine organisms,
डॉक्टर साहब आपकी पीपीटी और ये सब पोज हो गया है, कुछ भी सुनाई नहीं दे रहा है।
Okay, here it is fused with the nitrogen of the C ring. Okay,
डॉक्टर साहब स्क्रीन शेयर कीजिए प्लीज।
sir, I have already shared screen.
अभी शेयर नहीं है सर।
Okay, sir. Yes, sir. Okay, so these are some beta-carbolene N-fused biologically active natural products. Okay, you can see canthinone. Here it is fused with the nitrogen of this B ring. Here this is homophosphatase fused with the nitrogen of the C ring. Similarly, these are the other natural products that are biologically active and display various kind of biological properties. Okay. These are some example of bis-carbolene derivatives where two beta-carbolene rings are fused with each other. You can see here. This is a paganumine. It was recently isolated natural product and so anti-cancer properties with IC50 around 5.8 micromolar. So now coming to our research work, you can see this is the Cumigian C, or you can say 1,4-mile 9H beta-carbolene. It is a versatile precursor. Why? Because it have a 4-mile functionality at the C1 position, and there are two nitrogens that are basic in nature and nucleophilic in nature. Okay, one electrophilic site is present in close proximity with the two nucleophilic sites. Okay, so it is a trifunctional substrate basically, one electrophilic site and two nucleophilic site. Another thing, it is a you can say fast reacting electrophile also, because this ring is heteroaromatic ring. This nitrogen deactivate this ring, so this 4-mile group is strongly electrophilic in nature. Okay, so due to the presence of these two nucleophilic site and a electrophilic site, we can explore this natural product in a regioselective and chemoselective manner. We can block this nitrogen and selectively we can functionalize this molecule at C1 position. Alternatively, we can achieve cyclization through C1 9 position and C1 N2 position. Okay. So first of all, you know that natural products, isolation of natural products from natural sources is a tough process. Okay, because their concentration varies with the season, varies with the region also. So we have synthesized a gram scale procedure for the synthesis of this natural product, and that is also an economic way. We conducted the Pictet-Spengler reaction of L-tryptophan with dimethoxyacetaldehyde in 10% TFA that furnished this tetrahydro beta-carbolene acetal moiety. And further, we achieved oxidation of this C ring with N-chlorosuccinamide in DMF. During this procedure, we observed that along with oxidation of this C ring, decarboxylation also occurred. Okay. Then demasking of this acetal was achieved with aqueous acetic acid, and that furnished this Cumigian C in more than 10 gram scale. Okay. The advantage of this procedure are, it is an economically viable procedure. High yields are obtained at each step, and no column chromatographic purification is required at any stage. Okay, within 3 steps we can achieve this natural product. Then we explored the synthetic application of this natural product, and we treated it with various amino acids. When it was treated with L-phenylglycine in presence of iodine in sodium bicarbonate, decarboxylative amination occur, and we obtained beta-carbolene N-fused imidazole derivatives. Okay. And the structure of these products was confirmed on the basis of NMR spectroscopy as well as single crystal data. Then we explored the scope of this reaction with various amino acids like glycine, alanine, serine, valine, containing the aliphatic substituents in presence of iodine in sodium bicarbonate. And at room temperature or at 60 degree, this reaction was obtained completed within 3 to 7 hours and afforded the products in very good yields. Interestingly, when this reaction was conducted with L-serine, which is a bifunctional amino acid, we observed that along with formation of beta-carboline N-fused imidazole, intramolecular esterification reaction also occurs, okay, to afford this pentacyclic framework. Okay, you can see there are 5 rings that are fused with each other. Okay, so this was the library of compounds that were generated, and we also found that these compounds were fluorescent in nature, and we investigated their light emitting properties and found that these compounds exhibited quantum yield up to 50%. This is the proposed mechanism for the formation of these beta-carboline N-fused imidazole derivatives. Initially, this chemical undergo condensation with alpha amino acids with the loss of water, and this reaction is assisted by the iodine to give this imine intermediate. Then it reacts with iodine again, and an iodonium intermediate is formed. Then this base help in the process of decarboxylation. Okay, and this carboxylic group is lost as a CO2, and it results in formation of this azomethane ylide intermediate. Okay, which rearranges, and then it undergo 6-pi electrocyclization. Okay, to generate this beta-carboline N-fused imidazole derivatives. Okay, in another study, we achieved the synthesis of beta-carboline and butyrolactone based molecular hybrid. You can see that beta-carboline as well as butyrolactone is found in vast number of natural products. Okay, so we generated a molecular hybrid of these two pharmacophores. Okay, for this purpose, we reacted this 1,4-mile beta-carboline with this allyl bromides or cinnamyl bromides in presence of indium in THF water. It was found that when the reaction was carried out under semi-aqueous condition, we directly obtained the cyclic product. Okay, this intermediate was formed, but it promptly undergo intramolecular cyclization within one step to furnish these beta-carboline lactone derivatives. It was observed that water play important role in this reaction. When we used freshly distilled anhydrous THF, reaction don't occur. Okay, and another role of water was that it was responsible for the trans-stereochemistry. Okay, generally lactones are obtained under Barbier type condition with cis stereochemistry, but we obtained the product with trans-stereochemistry, and that was also confirmed on the basis of single crystal data. Okay, these are the transition states that are proposed. It was found that water have a very important role to play along with this nitrogen. Nitrogen of the C ring have important role to play. Okay, water play dual role. It activates, it polarizes this OH. Okay, making it more nucleophilic. At the same time, coordinate with the ester, making it more electrophilic. Okay, so dual activation is achieved with the help of water, and we generated a library of these 28 compounds in very good yields. Okay, in next study, we were interested to generate a natural product. Okay, so for that purpose, we reacted 1,4-mile beta-carboline with nitromethane by using ammonium acetate just as a base and acetic acid as a solvent. Okay, we were anticipating this product. This was our expected product, a nitroalkene. Okay, and after the formation of this nitroalkene, we were interested to react it with cycloalkenone. Okay, to give this type of Michael adduct. Then reductive cyclization and oxidation to generate analogues of eudiostomine U. That is a indole and beta carbolin based natural product. Okay, this was our research plan that we were interested. Okay, but when we reacted these molecules, nitromethane, ammonium acetate in acetic acid, we ended with this product, this carbolin derivative. Okay, and a heterosynthetic analysis revealed that two molecules of 1,4-mile beta carbolin undergo condensation with a molecule of ammonia. Okay, then we optimized the reaction conditions, and we found that when we use 2.5 equivalent of ammonium acetate in acetic acid at room temperature, within 2 to 4 hours we obtained these products in excellent yields. And there was no role of nitromethane actually. It at all not participated in the reaction. Okay. Then we also carried out the cross condensation reaction between 1,4-mile beta carbolin and other aromatic aldehydes. Okay. There we obtained the desired product, but it was found that aromatic aldehydes, which were more electrophilic than 1,4-mile beta carbolin, only can undergo cross condensation with 1,4-mile beta carbolin and ammonium acetate. And this product was obtained always as a minor product. Okay. The scope of reaction was also investigated with aliphatic amines, and we generated this formyl functionalized beta carbolin N-fused imidazole derivative. Okay. Because these molecules were highly conjugated, so these were also found to be fluorescent in nature, and we investigated their fluorescence properties, and we found that a quantum yield up to 26% was obtained. Okay. In another study, we tried to explore the 1,3 dipolar cycloaddition chemistry, where first 1,4-mile beta carbolin was subjected to Wittig reaction to generate this beta carbolin linked activated alkene, and then these were reacted with nitrile oxides that were generated in situ at -78, and within 3 to 7 hours it reacted to generate the diastereomers of beta carbolin C1 linked isoxazoline derivatives. Further oxidation of these isoxazoline with KMNO4 furnished the beta carbolin linked isoxazole derivatives, and these products were obtained in excellent yield, 76 to 92% yield. Okay, so we tried to extend the scope of this strategy for reaction with nitrile amines. Okay, nitrile amines, but we found that reaction does not work in a very good manner, and the product was obtained just in 16% yield. Reaction was very poor. Okay. Then we generated beta carbolin linked enones, okay, as a precursor, and reacted with nitrile oxides. In the same way, we obtained beta carbolin C1 linked isoxazole derivatives in a highly regioselective manner. Okay, high regioselectivity was obtained, and the stereochemistry was confirmed with the help of single crystal X-ray data. Okay. In another approach, we generated beta carbolin isoxazole C3 linked beta carbolin derivatives. They are C5, but these are C3, where we reacted this 1,4-mile beta carbolin with hydroxylamine to generate their oxime, then reacted with NCS to generate their chlorooxyamyl chloride, then in situ generated their nitrile oxide and reacted with alkyne, terminal alkynes like phenylacetylene. Okay, and we obtained these isoxazole C3 substituted beta carbolin derivatives. Okay, as we had just seen that we obtained beta carbolin linked pyrazole derivatives in very poor yield by using nitrilamines as the intermediate. Okay, so we thought of a different method where we reacted these enones, beta carbolin linked enones, or you can say beta carbolin linked chalcones, with the hydrogen hydrate in acetic acid. Okay, within one step we obtained these these pyrazolene derivatives within 45 minutes and in excellent yield. But it was observed that in acetic acid and acetylation also happened simultaneously. Okay, to generate the pyrazolene derivatives devoid of this acetyl moiety. Okay, we followed another method where they were reacted with hydrogen hydrate and iodine as a catalyst, iodine DMSO as a catalyst. Okay, and in this case, within one step, we obtained the pyrazole derivatives. Oxidation of this pyrazolene ring also happens, and we got directly obtained the pyrazole derivatives. Okay, and this was the small library of beta-carboline linked pyrazolene and pyrazole derivatives that were prepared by using this strategy. Okay. In another study, we demonstrated the versatility of this substrate for multi-component reactions, where it was reacted with 2 aminopyridines and isocyanides and 2 aminopyrimidine derivatives, where beta-carboline C1 linked imidazopyridines and imidazopyrimidine derivative, piperazine derivatives were obtained. Okay. And this is the proposed mechanism for first 1,4-methyl beta-carboline react with 2 aminopyridine to give the imine intermediate. This reaction is assisted by the indium triplet. Okay, that was used as a catalyst. After that, it undergo 4+1 cycloaddition with the isonitriles, okay, to generate this intermediate. Then it undergo 1,3 hydrogen shift to give beta-carboline C1 linked imidazopyridine derivatives. Okay, and this was the library of 29 compounds that was generated by using this methodology. Okay. In another study, we used zinc oxide based nanoparticles as a Lewis acid catalyst for synthesis of beta-carboline C1 linked benz-imidazole derivative. Okay, where 1,4-methyl beta-carboline react with the orthophenylene diamine in presence of zinc oxide nanoparticles. Okay, and we easily obtained these beta-carboline C1 linked benz-imidazole derivatives. The strategy was also explored for the synthesis of beta-carboline C1 linked benzothiazole and benzoxazole derivatives. Okay, but you can see while the synthesis of beta-carboline linked benz-imidazole derivatives, we have a lot of commercially available orthophenylene diamines, but derivatives of thiol derivatives are not available. Okay. So the limitation with this strategy remained that we could not introduce diversity here in the benzothiazole ring. Okay. It was found that these benzimidazole and benzothiazole derivatives were also very fluorescent in nature, and benzothiazole displayed the maximum fluorescence with quantum yield up to 28%. And this was the library of compounds that was generated by using this methodology. Okay. As just we had seen that for the synthesis of benzothiazole derivatives, 2-aminothiophenol based precursors are not commercially available, or they are very, very costly. Okay. So we tried to develop another methodology where we generated a sulfur insertion approach, iodine assisted sulfur insertion approach. Okay, where we reacted 1,4-methyl beta-carboline with the aminocarbazole derivatives in presence of elemental sulfur. You know, elemental sulfur is a very, very cheap reagent. Around 500 gram cost just 450 rupees. Okay, and it has been found that sulfur is a stable molecule, but it can be very, very easily activated just by temperature, just by using the polar solvent, various bases or various Lewis acid or Lewis base. It can be easily activated. Even silver, gold, or palladium catalyst react with the sulfur, okay, and they cause the activation of sulfur. Instead, the catalyst quenching happens, okay. So in this case, we obtained the sulfur insertion products, and these annulated benzothiazole derivatives are obtained. When naphthylamine was obtained, again CS insertion, carbon sulfur bond formation along with intramolecular cyclization happened, and we obtained the desired products. Similarly, with various anilines that were electron rich in nature, easily sulfur insertion happened in presence of iodine in DMSO, and we obtained a library of beta-carboline C1 linked benzothiazole derivatives, specifically with lot of diversity in this benzothiazole ring. Okay. It was found that these compounds were also highly fluorescent in nature, and in this case, quantum yield up to 92% was obtained by using the quinine sulfate as a standard. Okay, you can see they were the blue light emitter. Okay, so with these previous results, we were highly active. We were highly inspired to use the sulfur-based reactions because the sulfur can be easily activated. Okay, so we again explored this sulfur insertion methodology, and we found that even triethylamine-based base can activate sulfur, and we can achieve denitration strategy, denitration also. Therefore, first we reacted 1,4-mile beta-carboline with the 2-nitroacetophenone derivatives, where these chalcones were obtained. Okay. When they were treated with sulfur, elemental sulfur in presence of triethylamine, we obtained that insertion of sulfur at sp2ch bond along with denitration occurs. Okay. And in this case, triethylamine as well as DMSO serve as an activator. Okay. So in this strategy, one carbon-carbon along with the 2 carbon-sulfur bond formation take place within a single operation, and there is very short reaction time, and the compound displayed excellent fluorescent properties. Okay. So this was the strategy. You can see within 20 minutes at 60 degrees, this reaction occurs. Sulfur insertion as well as denitration occurs within 20 minutes, and products were obtained in very good yields. Okay. So this was the library of compounds that were generated. Various 1,4-mile beta-carboline derivatives and nitroacetophenone derivatives were used. Okay. Then we generated a one-port strategy where once they were reacted, this was formed, then methanol was evaporated, solvent, they were dissolved in the solvent DMSO, triethylamine and sulfur were added, and within one port this reaction happens. Okay. So basically it is a one-port, two-step procedure, and there is no need to isolate these chalcone derivatives, and directly from 1,4-mile beta-carboline, we obtain the beta-carboline C1 linked benzothiophenone derivatives. Okay. And these molecules again are very good, can serve as a very good intermediate. You can see they can serve as a good Michael acceptor. They can easily undergo 4+2 cycloaddition reactions and other kind of reactions. Okay. In another strategy, we generated beta-carboline and 1,3,4-oxidizole based molecular hybrids. Okay. In this strategy, this 1,4-mile beta-carboline was reacted with the hydrazides, aryl hydrazides, okay, and it resulted in formation of beta-carboline C1 linked 1,3,4-oxidizole derivatives. Okay. Again, iodine was used as a catalyst, cesium carbonate as a base. Okay. So you can see we have not used metal catalyst generally. There is another advantage that metal catalysts generally have some poisoning. They call when they go to the mainstream, they causes poisoning. In addition to that, they can bind with these. Yes, ma'am.
Virendra sir, I would request you to wind up in 5 minutes, please.
Okay, ma'am.
Yeah, thank you.
Okay. So this is the mechanism. Simply we get the hydrazone derivatives. Then they react with the iodine, with oxidative iodination happen, and a CO bond formation occur. Okay. So some control experiments were also conducted. In another study, we reacted 1,4-methyl beta-carboline with 2-amino pyridine derivatives, and sulfur activation was achieved, and beta-carboline based thiamide derivatives were obtained. Okay. This was the library of compounds, and in this case, we also developed analogs of LNGO-bucine, that is a natural product. Okay. So we also have used this sulfur insertion strategy for the synthesis of benzothiazole derivatives by carbon-carbon bond cleavage of isetine. Okay. We also have used this iodine catalyst for the A3 coupling for reaction of 1,4-methyl beta-carboline terminal alkynes and amines to generate these pyridocarbazole derivatives. Okay. And this is the library of compounds due to the shortage of time. And recently we have also synthesized beta-carboline C1 linked triazine derivatives by reacting with amidines. Okay. So overall, we have generated a variety of beta-carboline based heterocyclic frameworks by using a single precursor as a template. Okay. By using decarboxylative amination, 1,3-dipolar cycloaddition chemistry, sulfur insertion strategy, Barbier reaction, multi-component reaction, oxidative annulation, and oxidative amination strategies. Okay. So I would like to acknowledge the Vice Chancellor of Central University, our head of the department, funding agencies, CSIR and DST for funding our research program. And finally, thanks to all of you. Yes, if there is any question, you may please ask.
Anyone from the audience? Any questions, please? Any questions on the research work of Dr. Virender? Well, thank you, Dr. Virender, for sparing your valuable time to share your research work with us. We wish you all the best. May you come up with much more good work in future also. And with this, we'll wind up this session. We are running slightly late, but we'll meet at the scheduled time only. We'll meet back at 1:30 p.m. So session closes for lunch break, please. Thank you, everyone.
Okay, thank you.